one and all. I, Miss Pune and my co-host, Miss Richa Badke, take an immense pleasure to extend a very warm welcome on the behalf of Gokhale Education Societies, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, Nasik, on the occasion of 87th birthday celebration of Honorable Sir Dr. M.S. Gosavi, Sir. Today's international conference has been arranged entitled as Recent Innovations, a Holistic and Integrated Approach to Pharmaceutical Science.
As it is said and known that the value of an idea lies in the using of it, where creativity is thinking up new things, innovation is doing new things. Thereby, keeping in mind that there is always a way to do things or execute the ideas in a much better way, and as we speak about innovations and its recent advancements in pharmaceutical sciences, it is said that medicine is a science of uncertainty and an art of probability, where the overall association is restored for the improvement of the health status of the community by making a huge difference in the minds of people about the art of medicine.
In our glorious traditions, any program begins with a tribute to Goddess Saraswati. Saraswati, the goddess of knowledge and arts, represents the free flow of wisdom, consciousness, intellect, alertness, purity, and sattva-gun. I request Ms. Sakshi Deshpande to please begin the ceremony with Saraswati Vandan.
Thank you everyone.
I would like to request everyone to please rise and pay respect to our society.
chief guest for today's conference, Dr. Kanchan Bose, Director, Research Publication, Royal Institute of Management and Technology from Noida. Welcome, ma'am. We are honored to have with us the President of today's conference, Honorable Sir Dr. M.S. Gosavi Sir, Secretary and Director General, Gokhale Education Society. Welcome, sir. We are honored to have with us our Principal, Dr. S.V. Amrutkar Sir, Gokhale Education Society, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, Nashik, and convener of our 2 days international conference.
We have with us Dr. Vidu A.D. Nano, Professor and Dean, School of Pharmaceutical and Research, Jamia Mumbai, New Delhi, as the resource person. Welcome, ma'am. Today, we have with us Dr. Mridula Vedi Madam, NDMBP's College of Pharmacy, Nashik, as our session chair for today's conference. I welcome you, ma'am.
Also welcome all the respected principals and the faculty members and participants from various Now I would like to request our respected principal sir to please deliver the welcome address.
Hello. A very good morning and warm welcome to Honorable Sir Dr. M.S. Gosavi Sir, President of today's function, Dr. Kanchan Kohli Madam, Chief Guest of today's function, Dr. Vidu A.D. Madam, Eminent Speaker, Dr. Jaikant, Dr. Mridula Bole, all delegates and my colleagues. Today we are gathered here to have a 2 days international conference on recent innovation, holistic and integrated approach to pharmaceutical science on occasion of 87th birthday of our Honorable Sir Dr. M.S. Gosavi Sir, Secretary and Director General, Gokhale Education Society. Gokhale Education Society was established in 1918 and we have completed 104th year. We have 140 institutes in 3 different zones of Maharashtra, Mumbai, Palghar and Thane. Our all art, commerce, science colleges are NAC accredited and our HPT RYK Science College is going to complete 100 years in next year. We have schools, English medium schools, Marathi medium schools, then Adivasi Ashram Shalas. As a professional education concern, our society is a pioneer to start first university MBA program in 1968 with the leader of Sir Dr. M.S. Gosavi, and that was guided by Honorable J.R.G. Tata and Kirloskar Sir at that time. We have an engineering college established in 2009, then polytechnic colleges. As a health institute concern, we have a nursing colleges, physiotherapy college and this pharmacy college, and very soon we are planning to start medical program. We already started the hospital building, initially 106 bed hospital and then which could be extended to 600 bed hospital. That is in near future we may have a medical college in this campus. All these colleges are well known for their quality education, and our society is established by teacher, run by teacher, lead by teacher, and that's why there is always appreciation given to teacher efforts and student efforts. We do provide quality education at a very affordable cost. I would like to mention here my fee in a pharmacy college is the lowest fee among all pharmacy colleges in around Nasik. Similarly, we are providing a very low cost education to engineering. In the same way, we may provide education to medical students at a very affordable cost. And that's why our society slogan is Vidyarthi Devo Bhava. Now this college, after the name of our Honorable Sir, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, was established in 2012. We have just completed 10th year, but during this 10th year, we have started with the B.Pharm program, then we have added D.Pharm, then M.Pharm, and just recently we got a permission from Savitribai Phule Pune University to start PhD program. So gradually we are increasing our strength, and in the near future we would like to start additional PG programs and research laboratory which could be helpful for small scale industries around Nasik. So that is our planning. We are providing everything which student need as an infrastructure concern or a laboratory instrument concern. We have full-fledged instrument laboratory and research laboratory with all equipments necessary for formulations and chemistry point of view. Not only that, we do promote our students to participate in different events, national, international conferences, and our students have participated not only in India but outside India also. Some students have participated. Among that, Miss Radhika Ugle, she completed 6 months program at Prin, Italy and completed her graduation program there. Not only students but our faculty also taking initiative and participating in different events.
My 6 staff members have visited 6 different countries to attend international conferences. Not only curricular but co-curricular and extracurricular activity, our students are also doing very well. One of the students, she is a national basketball player. And as a co-curricular activity concern, we do promote our students to participate in all seminar, conferences and competitive events. Just recently, this year, after the corona pandemic, our students stood first and second in Met Explorer model competition, in that research competition organized for undergraduate students, and both the prize grabbed by our students. And recently, our students stood first in a presentation competition organized at Mumbai. So we are promoting all our B.Pharm students to participate, and not only that, we do also organize national and international conference since inception of this college, 2012. Initially, on occasion of our Honorable Sir's birthday, we did organize student-centric seminar, gradually to state level, then national level, and today we have our international level program. During COVID-19 pandemic also, we didn't stop. Ours is the first college to initiative and organize 1 week FDP program for teachers. 18 speakers were there, not from local but global. Out of that, 8 speakers from different parts of the globe, USA, Germany, then European countries, and that was a very huge program. And our activity was supported by Savitribai Pune University. And last year also we are organized in a national conference on occasion of this. Every year, this is from the inception of the college, we do organize 2 conferences, one in the month of September and one in the month of January. And in between, we do organize webinars every month for subject-wise for the students and staff. So we have been doing all this activity from last 10 years with the support of our Gokhale Education Society, and many times Savitribai Pune University also has supported. With the same, this year we have organized international conference on recent innovation, holistic and integrated approach to pharmaceutical sciences. Now, integration after National Education Policy 2020, everybody is talking about integration, integrated program. In the same way, with the theme, we have chosen this theme, integrated program.
As a innovation concern in pharmaceutical science, our convention additional dosage from tablet, capsules and modification of molecule. But beyond that we have to think now. Now because there is an integration of engineering and health
science has to be integrated, and many recent approaches like machine learning, artificial intelligence need to be used in a drug discovery and manufacturing processes.
Because as Corona has taught us, we need a urgent medication, fast development of the drug molecules, and because of this genome science, we could
Within 8-10 months, we could develop the corona vaccine. That is because of engineering technology merged with the pharmaceutical sciences, and many new innovations are going on. Among that, biodegradable reactors, because those who work in pharmaceutical industry know that validation and cleaning of all these reactors is a very tedious process, and that's why the engineering has helped us to develop the biodegradable reactors. And not only that, there will be a precision medication. This is a new, within a 10-15 years, we may have to face this issue, precision medication.
every individual will
A
have
separate drug according to his need, and at that time technology will help us exactly the cancers or other disorders, among that diabetes, then heart related, blood pressure, that may be a genetic disorder, and which genome or which genetic material is missing, that we could identify, and accordingly we have to develop the drug. And that's why 3D printing is the new innovation in this, and Spiritum is a new antiepileptic drug, already is in a process, get approval from FDA, and very soon it may come in a market. That is 3D printing drugs, so in terms of very huge manufacturing processes, the personalized medication, we have to print as per the requirement, print the drug and give to the drug. This may happen within a 10-15 years because there is a rapid development in engineering technology, and that's why it is a need to have a additional knowledge. Along with the pharmaceutical sciences, we must be capture the knowledge of engineering, mainly in a computer-related diet. Among the data analysts, the very huge requirement is there in the market, and all we know that we have to go for a clinical trials before introducing the drug in a molecule, and very huge data is required, and the data has to be analyzed. Engineering will do that, but if a pharmacy student is there, definitely that could be a great help for pharmaceutical industry to analyze the data, and clinical trials are going in a very huge, there is a huge market for clinical trials. Now students are getting opted for a clinical trial, and many software industries like TCS, Infosys, they are recruiting pharmacy students, BPharm and MPharm students for a data analyst. So that is a new avenue for the students of pharmaceutical science. But not only pharmacy knowledge is important, along with that, we have to have a knowledge of this computer related or engineering related knowledge, and that could be a great help and for you people to get a job in pharmaceutical industry. Within a 15 to 20 years, the conventional industry may get changed, and so we have to be ready for that. Not only this engineering and pharmacy knowledge is must, we have to have collaborated with the psychological and other things also, because during this corona pandemic, you might have faced, you might have suffered, or you might have heard many stories. People have suffered not only because of corona, but because of this fear of this corona. And at that time, many pharmacy graduates, postgraduates and people did help the people, need not to get worried. We have a medication, simple medications, paracetamol and other medications could help. And accordingly, there were so many stories had come in a newspaper, how pharmacists played important role in this corona pandemic, not only providing medication, but in psychological help to the students. So it is also need the patient counseling, one of the again major opportunity we may have in near future, patient counseling, because at least in India, doctors don't have time to talk much more. So we could play that role and to reduce the panicness among the students. because so many diseases and disorders are emerging. Among that, cancer now is like a temperature. Very out of 10 people or 15 people, we know somebody is suffering cancer. So that may be a genetic or some other external features may be responsible. Among that, advertisement is for cigarette smoking and all these things. So we could also help for people, give awareness about this, how this could affect on your health and why to avoid all these things. So these are all avenues is there, and that's why integrated approach is needed, and that's why our government has taken initiative to have an integrated approach. Not only music learning but psychological engineering, all this should be integrated, and you also can grab this knowledge during our 4-year or 4-year degree program, 2-year master program. Nobody is restricting us to grab the knowledge of other faculties, and I hope definitely you people can learn from this 2 days national conference. We have a very eminent speakers are there. They will share their expertise with you people, and definitely this conference will be fruitful for you people, and I am very sure that definitely this knowledge will enrich in pharmaceutical sciences. And for that, we have very eminent personalities today with us. Among that, the chief guest, Dr. Kanchan Kohli. He had accepted our invitation and she had come to our college. Dr. Kanchan Kohli Madam is a Director Research and Publication, Lloyd Institute of Management and Technology, Noida, UP. Madam is a M.Pharm PhD from very well-known Jamia Hamdard University, Delhi. Madam is having very huge 37 years of teaching and research experience, and she retired as a HOD of Pharmaceutical from Jamia Hamdard. She is a dynamic resource person, grab many projects from AICTE, DST, ICMR and UGC. Madam research profile mainly in herbal nano formulation. Now, nanotechnology is very, very important, and that's why today's lecture will be very helpful for you. Nano formulations in phytopharmaceuticals, cosmeceuticals and pharmaceutical domain. Madam has a project on curcumin based nano product in the market. It is already come in the market, SNSC 30. It is an emergency kit for DRDO. Madam is having a very huge publication. 200 papers have been published by Madam. Among that, 2 Indian patent, 1 US patent. Very huge citation in a research, 8,675 citations. HI index is 44, ITER index is 138, and she has supervised 45 M.Pharm and 25 students have completed PhD under the guidance of Madam. And with all these efforts recognized by Dr. Manjushree Pal Memorial Award 2020, Best Pharmaceutical Scientist at JSCC Mysore on 2nd September 2022 by APTI India. Madam is also got an PG Research Award from UGC, Best Project Award, Honorable Vice President of India, Technology Day 2003, IPA SCG SciTech Innovation Award, 69th Indian Pharmaceutical Conference, and IPGA Fellowship Award, Contribution in Pharmacy Profession. I welcome you, Madam, for this. For this function, we have a President, our Honorable Sir, Dr. M.L. Gosavi Sir, Secretary and Director General, Gokhale Education Society. Sir stood first class first in all his examination, whatever examination he appeared, BCom, M.Com, BA, LLB. Sir is the first Sahityacharya from Maharashtra Sahitya Parishad with first class first. Sir is the first PhD in Business Administration in India from Pune University in 1968 under the guidance of Dr. J.R.D. Tata. He is the youngest principal with longest tenure at age of 23, and he served as a principal for 37 years, and he has given his dedicated service to education for long 65 years. Sir is a life member of Gokhale Education Society since 1960, Chairman Business Administration India. He was the Dean Faculty of Commerce, Outstanding Publication Management Gap in Developing Economy. Sir was the Chairperson All Asia Management Conference. He received a Silver Memento Award by A.S. Sharif, Governor of Maharashtra. He started achieving excellent publication at the hand of Sri Shankarullah Kirloskar in 1988. International Award for Making University Education Entrepreneurial at University Netherlands 1992. Sir got a Rajiv Gandhi Peace Award, Nan Hira Award, Master Teacher, Millennium Award, Chancellor of International Association of Educators for work is Man of the Year, the Contemporary Who's Who American Biographical Institute, Knight of the Millennium Sir title at the hands of Sheriff Mumbai. Sir got Gold Medal Award, ICME. He was the President Maharashtra. He is the President of Maharashtra Council of Educational Administration and Management since 2006. Sir authored 20 books, over 100 research papers. His authority on management, thoughts for Vedas, Upanishads, Gita. Sir received many awards, among that Nashik Bhushan Award, then Sri Vidya Saraswati Awards at the hands of Bharat Ratna Lata Mangeshkar, Honorable Sri Yeshwantrao Chavan, Maharashtra Bhushan Award, APJ Abdul Kalam Gold Medal Award, Mahatma Gandhi Award, felicitated with Samman Patra at the hands of Sri Vidyasagar Rao, Governor of Maharashtra, Mahatma Gandhi Peace Award, Best Institution Management Award by International Achiever Conference at Bangkok, and Lifetime Achievement Award by Savitribai Phule, Pune University in 2020. I welcome you, sir. Thank you very much.
Now, as we move forward for the felicitation of our chief guest, I would like to request Honorable Sir Dr. M.S. Gosavi sir to kindly felicitate our today's chief guest, Dr. Kanchan Kohli, with a bouquet shawl and token of gratitude. Thank you, sir. Now I would like to request Dr. Kanchan Kohli madam to kindly felicitate. Thank you, sir. Now I would like to request Dr. Kanchan Kohli madam to kindly felicitate Honorable Sir Dr. M.S. Gosavi sir with a bouquet, shawl and token of gratitude.
I would like to request Dr. Kanchan Kohli ma'am to kindly felicitate Dr. S.V. Ambedkar sir. Thank you, ma'am.
I request Dr. Shilpa Haran ma'am to kindly welcome all the dignitaries of the dance.
Thank you, ma'am. Now, I request our chief guest, Dr. Kanchan Kohli ma'am, to address the gathering. Please, ma'am.
Pleasant good morning to all. Honorable Dr. Gosavi, Dr. Ramkar, and first of all, I'd like to wish Dr. Gosavi on his 87th birthday. Sir, happy birthday to you. I really, dear students and all dignitaries of the dais, on the dais, and good morning to all dear students, and I'm really very happy to be over here as a guest or with you people, and I'd like to share my views about pharmacy afterwards. And I came to know the vision which is written over here, and first of all, very happy with the progress of the Gokhale Education Society, and the first institute was started in 1958, and sir was the principal of that institute, and really motivated today by his progress and what he has told just in a minute, that every year he was adding one of the institute. So see his vision and how now you are finding all education wise, all your programs are there. So with the this motivation, I've been really motivated, and I think when you are listening these things, you must be all be motivated today that what will be your mission in life and what will be your vision in life. So what the vision of this institute I have seen to excel in pharmaceutical education and technology. And today what I am watching it, that it's there. And mission is to provide high quality pharmacy education and training to explore the student to be a responsible for professional pharmacist. And that's been, I'm watching it and I've been listening from the seniors, teachers in just in 5 minutes, and I came to know that how progressive this institute is under the guidance of Dr. Gosavi. So with this mission, which when the institute and the society is working, and then there is no length where we can progress. And very rightly, Dr. Amrutकर has mentioned about the artificial intelligence, and where I think you need a collaboration of engineering people. They are very well educated in computer program, and with this collaboration, I think you can do very well in drug discovery, and where many of the alumni students, old students, they can be accommodated in that program. So that artificial, if you will start with the artificial intelligence in drug discovery today, I think you can do wonders. And with drug discovery, as we know, that the drug discovery, what our computer system and what our system says, that initially you can go ahead with the research program. What you want, you just put a, like you want to work on a one plant, say for example, you just give one example of the plant, and today we are finding that our scientists, they have discovered plant in a such way that with the single plant you can have the many of the pharmacological activities. Just see the example of curcumin. So curcumin is, we all know that it is anti-inflammatory, it is Alzheimer's also, it is for we can say pains, joint pains, whatnot, cancer also, little bit activity is there. Of course, today we are saying we cannot replace anti-cancer drug with curcumin, but at least for the better management of the cancer patient's life, if you just substitute one capsule a day, That's what we have discovered, that's the nano formulation. You know why? What's the use of nano formulation? First of all, thing is there. I mean, whatever nano formulations we are doing, after all, I mean, they've been discovered, they've been done, lot of pharmacological studies. After that, we will be launching the product into the market. Just see the example of amphotericin, for example. At the COVID time, when the black fungus cases were there, you were finding the amphotericin formulation was in shortage. So could you tell me why it was the shortage? First thing was the raw material. That's the other thing. But why it was shortage? Why the shortage of amphotericin was there in the market? The reason was we didn't have technical staff to go for that formulation. Because when we had two types of the formulation, amphotericin. One is the plain as such. Another one is the liposomes preparation. So everybody was after the liposome preparation. At that particular time when the lockdown was there, everywhere a crisis was there, no manpower was there. So to develop this formulation was really difficult. And that time what had happened, even I had given counseling and I had given advice to one of the industry that please, sitting at home, please go the formulation of amphotericin this way. I had given some kind of the hints to them, and that time particularly those polymers were not there, those surfactants was not there, and it was really difficult for the pharmaceutical industry. But anyway, our pharmaceutical industry, they have worked, I mean, very hard to get these products into the market. And as we know, the shortage was there. Shortage was for the liposome preparation, not for the plain preparation. The reason was when we are going for the nano formulation, then we are improving the bioavailability of the drug. Means the drug which has to reach to the blood. Okay. Similarly, if you just see the example of plain curcumin and nano curcumin, and we had seen when we are giving the plain curcumin and doctor was just prescribing 1 to 2 grams of the curcumin per day in divided doses. So when we have developed the nano formulation, just see 30 milligram of the dose we took, and we could able to manage the bioequivalence study with the 200, even the 200 milligrams, 2 grams of the drug was not giving that result. The reason was that when we are giving the curcumin as such, no bioavailability was there. The drug was stuck to one place only at the stomach region. We have done the gamma scintigraphy study, and we have seen our gamma scintigraphy study. It is just reaching to whole of the vital organs like lungs, liver, kidney, all the vital organs, and then might be reaching to the brain also. That we have not explored much because we didn't have those many resources. Whatever the resources we had worked, and after working hard so much, then we have got it done bioequivalence studies from scheduled by the labs, and that's the preclinical studies on the animals, because whatever the animal studies we are doing in our lab, that's not acceptable to our Ayush Ministry as well as to the DCGI office. They are not accepting those studies because they are not as per GLP. So for carrying those type of the studies, we must go for the GLP. So this was the difference between amphotericin plain and amphotericin liposomes. So if we work with this mission and vision, that requires hard working, communication skill, and moreover, everybody has to work with honesty. Whatever the results you are getting, you should present in a true way. No manipulation. Sometimes I am finding the students, when they want to show the bioavailability enhancement, they will show as such without doing much of the work. So my advice is that everybody has to do work with honesty. If honesty is there, we cannot move anywhere. So that is the one thing which I like to mention to the young generation. Sorry. And we are not too fast to achieve the things. Maybe in a slow manner, you can achieve many things. As we are seeing today, the world is moving very fast, so everybody is after money. Okay? So what I am finding with the young generation, immediately they want a jump of 20,000, 30,000, but they will not stick to that place, but they will just, when they will be joining one organization, always that one thing is there, that how can we jump to the other organization? What height we will be getting? Maybe for 5,000 they will run, maybe for 10,000, without thinking what they have to do and what is their mission. So money is not everything in life. Of course, requirement is there. I am not saying you should not go after money, but slow process should be there. That jumping from one industry to another is not much advisable. That's the one thing I like to mention. Then, because for that reason, we cannot work in a honest manner. We have to just give the results and move to the other industry. So that vision should not be there. And we have to work with a the professional way, with a very honest way, and that's my advice to the young generation, that how they can move ahead. And you see, whatever achievement we have done, not in a single day. So I served for 42 years in teaching and research, and maybe in 2014 we would able to launch our product. And that's the one of the product whichever university, at the university level, the product has been launched into the market, and today if you will just Google it, you will get the Snack 30. And it's available at Amazon also. So just for fun joke, we have filed a US patent, thinking that how we can get a US patent is very difficult. No? And I was not aware really that we will be going to get it, and I have given all the rights to the industry. Because that time that awareness was not there. And that was the first patent which I filed at US patent level, and then we have spent a lot of money. Of course, that money was from the project itself. I have not spent from my own pocket. And then we have given all the rights to the industry कि हो गया तो आप product launch कर देना, नहीं तो ठीक है, क्योंकि हमें पता था patent नहीं मिलेगा. So luckily, within span of 2 years, we have filed the patent in 2011 in September, and 2014 we have got the patent. And then in 2016, the product was optimized to industry level, and then 2018 it was launched in the market, and during COVID time, much of the demand of this product was there. And see one curcumin, and if you will just see the papers of research, papers of the curcumin, at least you will be getting, when we have submitted the project, at least 2,000 papers were there. And when we were discussing with my team, with the students, scholars, they were saying, "Madam, इसमें तो कुछ नहीं मिलने वाला, क्योंकि already 2,000 papers are there. What you will achieve from this research?" So I told them, नहीं, हम थोड़ा सा nano की तरफ काम करते हैं. So that was the way we have worked. And in 2010 we have worked for the nano formulation. And today many of the nano products are there, which the industry wants the consultancy. So far I have not started that work. Of course, we have the product of lycopene that we have isolated from tomato. Then we have worked for green tea. There's a constituent ECG, application. So many herbal products we have there, and 2 of ICMR patents have been already granted. So with this mission, I would like to mention you that you work with honesty and hard work. So that will always pay. So that's from my side, that advice is there for the young generation. Okay? So thanks for the patience listening. I don't know what's going on, otherwise I will give you much of the examples which I will quote you afterwards. Okay? Thank you so much.
Thank you, ma'am, for your valuable and encouraging words and your guidance related to nanotechnology. Definitely, the students will implement it in future. Now, I request Honorable Sir Dr. M.S. Gosavi, sir, to bless us with your valuable words. Please, sir.
At the very outset, I compliment and congratulate your principal for organizing this international conference and giving you an experience of learning on the self basis. The topic which has been chosen and the experts who have made it a point to be here for 2 days, 6 sessions, is an excellent addition to your knowledge and also learning. See, we learn every day through lectures and many other methods. But this is the best method of learning, that we organize a seminar, a conference, we select a topic, we bring the experts, we write papers on various subjects, and during the course of our meeting with the experts and listening to these people, we can have questions in our mind which can be answered by. This question answer method also is a very important part of this particular activity. I am very happy that Dr. Bansha, you are chief guest for this function, has given you a number of things. She has given the best advice that can be given on this occasion. Number 1 is, she said we have to be very honest. Honesty is the best policy. And you flourish and progress in life on the basis of honesty on one hand and your knowledge on the other. Nothing else is required. If you go on adding to your knowledge, you acquire in-depth knowledge. You acquire world knowledge available on the subject. And be faithful to what you are doing, as she said, honest. The honesty will help you to bring your own personality in the limelight. Secondly, she said, we have to be professional. It's a very important part of our life. We are here in the professional subject, learning that subject to become a professional. There are other conventional areas where students are also learning. But they lack this particular element which you are now going through. So this professional approach is very important. And last but not the least, this method of learning, seminars, conferences, small group discussions. So this develops your personality and make you update in your knowledge. You become little more bold to ask questions, to understand difficulties that we have while discussion. And we meet number of people. Learning individually and learning in a group and learning in a seminar has a great advantage. When one speaks, we all listen. And that's a great opportunity for all of us to immediately think over what has been told to us. And if there are any reactions, please make them. Please put up them. And see that all these things are clarified. Even if one student asks a question, you say all are benefited. So this is the beauty of this seminar and conference. Apart from that, I am very happy that Gokhale has organized this particular program on a subject where each one can study and learn and contribute. I remember when it was asked what is the best method of learning and understanding at graduate and postgraduate level. Because as you all know, in various faculties, at undergraduate level particularly and also at the postgraduate level, lecture is the only method of learning. And somebody comes and gives you a lecture, there are books also. And you have only two resources. One is reading the book as itself or listening to a lecture which lecturer is giving. But in a professional course, we have to be a little more practical. Along with theory, we must know the practice. And along with theory and practice, we must also know how we will be able to do best in a given situation. We are dealing with human beings and we are going to apply certain aspects of our knowledge for making their life more simplified, more healthy. And it is from that point of view that learning through this particular method for topics which are already there at the hands of the experts will go a long way in creating a proper personality which we should. We don't only have or we don't only want knowledge. We want the understanding of the subject. And it is through this method that we come to understand. I am very happy that we are in the pharmacy college and also we have started some other colleges and we are going to start a medical. Our idea is the students should get as pure knowledge as possible and as accurate knowledge as possible at the hands of the expert at least cost. For such program, if you want to go out, stay somewhere, pass your 2 years, 3 years, whatever the program is, come in contact with those people and learn. If there is no alternative, you have to do that. But why not find alternative? And we can also give quality and we will come up with quality. See, quality is a state of mind. If you think that this is to be done in this way, and I will see that I get the quality, I get 100 out of 100, it's not impossible. Therefore, a firm determination to take instructions, education and knowledge with a firm belief that one can become an expert in that particular line, one can become number one in that line. You can make the best of your time. And I am seeing for the last more than 10, 15 years, when we have started these colleges, engineering, pharmacy, and many other colleges starting with nursing and others. And now we want to start even a medical college, where the cost of education will be reduced. The quality of education will be heightened and students will be given enriching satisfaction that what they want is very near to them. And this is what we want. Otherwise also you can go out and bring so much of this knowledge and do something for your own self. But the real question is, has the society benefited? Has the society become little different than what it was? For 50 years we are giving this education. And what is our contribution to the society at large, to the common man at large? For bringing the health of the people little up and standards of living little different. So this is the question that we have. So we are taking education mainly for our own self. But that is of no use if I keep it to myself. We have to be very useful to the society. And for that purpose, learning at minimum cost, learning in a higher group, in a total group, the quality of education will change. And therefore we have made facilities available to the students in one campus, in different branches. And as I said, when we started with arts college, 100 years back, we started with an arts college. That itself was a great innovation. No college was there in this part of Maharashtra. And that day it was there. And at that time 100 students were, you see, on the roll in the very first year with one girl. So this is the starting point of Gokhale Education Society in higher education. Today we have 25,000 students. Half the number is of girls. And we are providing 13 faculty educational programs and giving to the students latest and best in the line. We are the first to start MBA program in India. The word MBA was not known. It was only known in Harvard and Stanford. And there was some effort made by Ahmedabad and Bombay International Institutes established by the government. But that was for the people who are already in the job. So those who are executives were brought together and given some training program for 2 days, 3 days, 4 days, like that. So that was the idea of business administration education of the government of India. We wanted fresh graduates to be prepared after your matriculation or SSC, what we call it. Why it is necessary to wait for such a long time for education which is in demand for all over the world? And believe me, you will be little different than others. Maybe that you will not be able to do what they are doing at that level. But education is to be constantly there for demolding as per requirements of the time. And we are the first in India to start this Master of Business Administration program as early as in 1966. 50 batches have gone. Every batch consists of 35 or 40 initially and then 60 students according to university norms. So many students have got opportunity to learn here. And I learn from them every day how best they are doing and how enriching satisfaction they have with the education. So you are therefore belonging to that generation where education has become diversified, education has become very, very practical and pragmatic, and you are given opportunity to a number of subjects. As rightly said by our chief guest, in this program, every student must be aware of what is artificial intelligence. Subjects like that which are coming, blockchain. Now these are the subjects which are the subjects of 21st century, second decade, third decade. And like that new subjects will come every decade. And we must be aware of that. So keeping ourselves updated and also learning the subject the way we should learn in a larger class, in a smaller group, and also with our friends, individually and in a small group. We can bring quality to education. Number of books have come to the library, and number of experts are coming to guide you for the program. This was also absent in the past. It was a full-time teaching job, and therefore a full-time teacher was the only concern. There are limitations. One can become an expert in a particular subject, but to bring the real field experience, I must also work there and bring that experience and put it before you. And who knows, anybody will benefit or somebody will benefit, if not all. So therefore this faculty from industries, we call it associate faculty, visiting faculty, contributory faculty. And any person can become a contributory faculty who has experience in that line. Age is not the parameter. This is your experience and your visitor will be the parameter. So if you bring before you the ramifications of the subject from different angles, from full time, from part time, from visiting faculty, from books, and from experiences in the seminar of the experts, you see, you are exposed to that knowledge in such a way that you become more perfect in the shortest possible time. And that is the purpose of giving education at a higher level, because you are already having a mastermind. According to me, every student has a mastermind after his graduation or up to graduation. And then there is question of self-learning and self-knowledge. But little help is necessary from outside, and that help we are providing in these programs. Therefore, education is to be made updated. As far as possible, we have to be very practical on the subject. We must learn in a group. Whatever we are learning, we must be able to put that for the use of the society at large and give the benefit to the community. If these are some of the aspects which are remembered by every student, I am sure you will get a new approach to the education that we are doing. And from this point of view, this seminar or a conference of this kind, when you are exposed to the experts, and you are in a larger group, you are in a smaller group, you read from the books, you understand from the teacher, this will go a long way in making your mind more stable, more silent, and quite quiet so far as that subject is concerned. So many aspects are there for every subject, there are a number of aspects. There is a physical aspect, there is a mental aspect, and so on and so forth. Similarly, there are other aspects also. Historical things are there, ecological things are there. So all these aspects can come in a seminar or conference that we organize of this type, and this is the great advantage that our learning has provided, learning institution has provided in the present time. I am also very happy that you are all participating in this particular program with best of your attention and also based on the services that you can give for this particular program. I therefore wish that in addition to the number of other programs that you get, lectures, tutorials, exercises, this method of learning will be added to the program in a meaningful way, and we make it a best method and regular method of learning. Our total learning becomes very, very impact, very, very good. Thank you very much.
Sir, for your inspirational and motivational words and guidance related to honesty, creativity, intellectual alertness, the key to professionalism and the process of learning, not only to acquire great knowledge, but to develop ourselves and excel with a sense of quality in our lives. I'm sure everyone present over here is included in another life. I request Dr. Prashant Phule sir to kindly deliver the vote of thanks.
Good morning, one and all. Gratitude is the most beautiful form of courtesy. It's my great privilege to propose the vote of thanks for inaugural function of 2 days international conference on recent innovations, a holistic and integrated approach to pharmaceutical sciences. I, on behalf of Gokhale Education Society, Sir Dr. M.S. Gosavi, Professor of Pharmaceutical Education and Research, and entire team of my college here today, and on my own behalf, extend a very heartfelt gratitude to Honorable Sir Dr. M.S. Gosavi Sir, Secretary, Gokhale Education Society, for being with us and accepting our invitation as a President for today's inaugural function of 2 days international conference. I sincerely thank you, sir, for enlightening the delegates with great words of experience, wisdom and motivation. Thank you, sir. I wish to acknowledge my deepest gratitude to Dr. Kanchan Kohli ma'am, Director Research and Publications, Lloyd Institute of Management and Technology, Noida, UP, for accepting our invitation as a chief guest for today's inaugural function. Madam, your 40 years of experience in research in pharmaceutical science will definitely motivate the delegates and young scientists of tomorrow. Thank you, madam. I am thankful to Dr. D.P. Deshpande ma'am, Director HR, for being always as a source of motivation with us. I would like to express my gratitude to Professor P.M. Deshpande sir, Director Project, Gokhale Education Society, for constant support in every event. I am thankful to our principal, Dr. S.V. Amrutkar sir, for motivation in all the activities conducted at this college. Dear delegates, we all need someone who inspires us to do better than we know how. I am thankful to our mentor, Sri Shailesh Gosavi sir, for his continuous motivation and support in every event of this college. I am thankful to today's speaker, Dr. Vidhu Eri madam and Dr. Jaikant Chandrasekharan sir, for accepting invitation as a resource person of today's session. I am thankful to Dr. Budula Bede madam, Dr. Rupali Patil madam and Dr. Sanjay Shivsagar sir, session chair of all three sessions of today's conference. I take this opportunity to thank Gokhale Education Society Nashik for providing financial assistance for this particular conference of 2 days. I am thankful to my team members, all teaching and non-teaching support staff members, for their support during this event. I am thankful to Praveen Thakre and his team for live streaming of this conference. I am thankful to all the delegates who attending this conference and patient listening. Last but not least, I am thankful to today's anchor for their fabulous anchoring, Ms. Rucha Dadke and Ms. Poonam Bagad. Thank you very much. Thank you.
The inaugural function is over. I request everyone to please be present as we start.
occupy the dais. Please maintain silence, everyone. Please be seated. Now, we are going to start with our first session entitled Paradigm Shift from Conventional to Innovative Pharmaceutical Research. For this session, we have with us Dr. Kanchan Kohli as the resource person and Dr. Mrudula Bere, NDMVP College of Pharmacy, as well as Pharmaceutical Development Scientist and Patent Consultant as our session chair. I request Dr. Shilpa Harak ma'am to please introduce Dr. Mrudula Bere ma'am. Please ma'am.
Thank you, Rucha. It's a great pleasure and honor to introduce my colleague and senior Dr. Mrudula Bere ma'am. She is a pharmaceutical development scientist. She has various consultancies in her name, and her base is, has Rucha already said, NDMVP's College of Pharmacy. Ma'am has done her B.Pharm, M.Pharm and PhD from NDMVP's College of Pharmacy. In her gate, which it was in 1995 when the pattern was totally different, she has had an all India rank of 26. So we have not only a technically but also academically meritorious person with us today. Ma'am has completed her PG diploma in IP law from Nalsar, Hyderabad in 2009, where she secured an all India rank of first. Then she has completed her LLB from MVP College of Law and LLM from University of Turin. She has a total experience of 20 years, all at NDMVP's College of Pharmacy. She is also a director in the pharma division of pallet advertising and multimedia. She worked there for 3 years. Has a patent. She is now a patent consultant and advisor and caters to organization within the country and abroad also. She is a volunteer at the Shatabdi Hospital Ethics Committee graphic in 2009. Ma'am has 3 patents to her credit. She is a regular column writer on IP governing pharmaceutical industry. She has received the Best Book Award 2017 from Maharashtra State Government, and she is the only Indian to get fellowship for LLM by WIPO and Japanese Patent Office. We welcome you, ma'am. Thank you.
Thank you, ma'am. I would like to request Dr. Kanchan Kohli, ma'am, to felicitate Session Chair Dr. Mrudula Bere, ma'am, with bouquet and shawl. Thank you, ma'am. I request Dr. Mrudula Bere, ma'am, to kindly introduce Dr. Kanchan Kohli, ma'am. Please, ma'am.
हाँ हो सक जस्ट मिनट आई विल ओपन now it is working thank you
Welcome. Thank you. So early good morning to you. And one thing I like to tell you that your principal has mentioned about the two programs. One was on the genomes and other was on the artificial intelligence. That time I couldn't able to speak, and I like to give you the collaboration, like in your university you are having both the things with you. You are having pharma background people also, your teachers, scholars, and you are having science section also. So why can't the programs on genomes could be started and the research work? I like to suggest you, not advise, but suggest you that you can start with that program on genomes and on artificial intelligence. Whereas for both the programs, you will be having the collaborations in your university only. You need not to go in anywhere. If you just talk with the engineering people, they will give you kind of the suggestion, and from pharma you will be having the drug discovery channel, and in both collaboration you can achieve like many things if you want. So this is again one of the suggestion from my side. So today's lecture is that's the paradigm shifts from conventional to innovative pharmaceutical research. As we know, now we are in the new era, so where we have to move from past to present, like from traditional to modern. That's what the requirement is there. We cannot, like as the technology is going into so much advances are going on, like today we are talking about the artificial intelligence, then the translational research, then we can say you're the computational program, then we are having kind of the targeted systems. So when these technologies advancements are there, then we cannot go behind like what the tradition was there. When the traditional knowledge was gathered from Vedas, Shastras, where things were written, and those things, that's what we are using, and from the ancient time people are using, not we as a pharmacist. When we have come to the pharmacy profession, then the things have been generated from past ancient time was there. Like I'll give you one example, like yoga. So yoga was there from the ancient time. It has not been, we can say the newer thing which has been added. But when you see, when the Ramdev has come to the picture, you are finding so many, the people been excited for this, and they have learned so many kriyas for this reason. And maybe in our family, children, elders, they've been all excited to go for the yoga programs. So this is from the, we can say, that was the traditional knowledge. And this is what he's teaching, the way he's teaching, and the way he's doing the practically. And he's teaching us practically, not the theoretically. So that's what the learning is there. So that's what I want we should achieve as a pharmacist all together. Of course, now I'm from the old generation, but as the new generation is coming up, so I like to advise you, please go for these programs, and you will see many findings you can achieve. And today I'm really excited for those programs. And maybe Dr. Amrutkar just has mentioned, so I've got little excited also. And last month only, I had a talk with engineering people, So they have come up with I said, how we can go for the artificial intelligence. So then he has told me this. So I told him that we can go for in silico drug discovery. So why not the collaborations could be done, and one can, today the word is not we can work in isolation. We have to work in all together. ऐसे पहले होता था कुछ लोग अपनी सम रिसर्च करते थे तो उसको छुपा लेते थे कि इट शुड बी हाइडेड, इसको नहीं बताना. On what I was doing the work, whatever platform I was getting, I was telling everyone to start with the kitchen masalas only. Start with the kitchen masalas and you can achieve wonder. And take one masala, जैसे जीरा है, काली मिर्ची है, कुछ भी ले लो, आपको सब प्रॉपर्टीज मिलेंगी. वाई कांट यू एक्सप्लोर देम? एंड कुछ करने को नहीं है, कुछ करने, पीपल आर सेइंग नथिंग इज लेफ्ट, एवरी ड्रग हैज़ बीन फिनिश्ड, उन्होंने इतना काम कर लिया, सबने नैनो पे काम कर लिया, सो वेयर इज़ द वर्क टू डू? But still, I'll tell you, lot of work has to be done. Even you are, I mean, whatever the number is sitting over here, if you just start doing for one constituent, and you can file many patents and see how the emotional excitements you will be having when you are achieving something. So that's why one of the advice which I wanted to give during inauguration time, but due to my voice, I don't know. So this is the lecture which I have chosen for the young generation particularly. And of course, my teacher colleagues also. I mean, I'm not giving such kind of the advices कि नहीं हम ऐसे करेंगे. But some suggestions are there. If you will take it, I think you can achieve something. So that's up to you. Even you want to take it. Okay. So then paradigm shifts from conventional to innovative research work. So drug discovery and development, this is the main thing. So just in the past, most drugs have been discovered either by identifying the active ingredients from traditional remedies or by serendipitous discovery. मतलब ऐसे ही किसी ने बता दिया कि this drug is having, जैसे हम लोग विलेज में गए फॉर एग्जाम्पल, और वी हैव गॉन टू द हिल्स, तो सम plants have not been explored over there, but they are doing it, they are using it, they don't know the botanical name, they don't know the chemical constituent, but they are saying कि this is which we are using for reducing the blood pressure. जब stomach में pain हो जाती है तो we are giving this medicine. If you just move to the hills, you will come to know so many plant materials which been unexplored. So why can't we bring one of the material from that place and with their advice, so we can start with that working. So, but now we know diseases are controlled at molecular and physiological level. That's what we were talking about, the genomes. Also shape of an molecule at atomic level is well understood, like we are having the softwares, drug discovery, in silico studies. So for every molecule, we can have the in silico studies initial before starting our actual research work. So what I do is, just for example, I'll give you the example, I have to work for hemp seed oil. I've started working on this for hemp seed oil, for example. If you will find in silico studies, it may contain your THC, that is having the psychotropic effect. It may contain CBD, that is very good for antidepressant. So why can't we always say that it's narcotic, but it is having medicinal properties? Unless until our government will not take those steps, will not come forward, but at least quietly for research purpose, nobody will tell you that not to go, because I have inquired from DCGI people, I have inquired from so many platforms, can we go for, although it is in the narcotic, but still we can go for the research purpose. And similarly, like hemp seed oil, it can be used externally also, internally also, and there are so many wonders are there. It contains omega-3, omega-6, omega-9s, so you can explore. Maybe the day will come, it's better than our almond oil. So if you find the cost wise, it is much cheaper. Only thing is our minds has not been set up that we can use the hemp seed oil and we can substitute almond oil with hemp seed oil, because it's one of the cheapest source and easily available also. If you will just search for Amazons and any other Flipkart, you will find so many companies are giving these hemp seed oils. So why not from the drug discovery, we can able to find out that what is the maximum activity it is having. For example, omega-3, 6, 9s are there. Why can't we explore for antioxidant properties? And when it is one good antioxidant molecule, naturally it will be having so many wonders like anti-cancer properties, and we can say even for weight management also. So many activities are there, but we have to select out of it what is the best. So best, either you have to go for the animal studies, but we want to avoid these animal studies. Why can't we have the drug discovery in silico studies? What our motto is there, and for that reason many softwares are there. We can use them and we can bring the molecule into the right track. Similarly, we are having the information of human genomes. The history of drug discovery, pre-1919, we have the herbal drugs. Again, the serpentarious discoveries, मतलब जैसे किसी ने बोल दिया कि ये ड्रग इस चीज़ के लिए बहुत अच्छी है, तो people started using of it. जैसे if you just ask your parents or forefathers, they will tell you कि हमें उसने बताया था कि अदरक खा लो, आपकी खांसी ठीक हो जाएगी, आपका गला ठीक हो जाएगा, आपका उल्टी बंद हो जाएगी. तो अब इसमें से दैट्स अप टू अस कि हमें क्या एक्सप्लोर करना है, उसको किस लेवल तक एक्सप्लोर करना है, कौन सी ड्रग डिलीवरी में जाना है. तो फार्मास्यूटिक्स इज़ नॉट दैट कि हमें ड्रग बनानी है, फॉर्मुलेशन करनी है, बट वी हैव टू थिंक कि क्या बनाना है, विच डोजेज फॉर्म एंड विच इज़ द डोजेज फॉर्म विच विल बी हैविंग द इंडस्ट्री एप्लीकेशन ऑल्सो. ये नहीं हम बना के एक थीसिस बना के रिसर्च पेपर बना दिया तो वी आर वेरी हैप्पी, हमारे 2 पेपर्स आ गए. यू नो, मैनी ऑफ द हिडन थिंग्स आर देयर विच येट टू बी पब्लिश्ड. तो मेरे को आई डोंट वांट कि हम इतना पब्लिकेशन कर दें. आई वांट कि आप उसकी इंडस्ट्रियल एप्लीकेशन देख के चूज़ करो. वेन एवर यू आर सेलेक्टिंग फॉर योर M.Pharm एंड PhD प्रोग्राम, प्लीज़ सी कि हमारा प्रोडक्ट जो है कोई ले ले, कोई इंडस्ट्री हमसे हमारी वैल्यू डाले, तो यू विल बी हैविंग योर ओन वैल्यू ऑल्सो एंड कम्युनिटी विल बी बेनिफिटेड एंड दैट पर्टिकुलर मसाला विल बी यूज्ड इन ए प्रॉपर वे. नहीं तो ऐसे भून भून के सब खराब हो जाता है ना, तो यू विल बी हैविंग द वॉलेटाइल कॉन्स्टिट्यूएंट से फिक्स्ड ऑयल तो चलो हम सेव कर लेते हैं, बट देयर आर अदर कॉन्स्टिट्यूएंट्स विच विल बी डिस्ट्रॉयड बाय स्ट्रॉन्ग हीट. इतना आप उसको हीट करते हैं, तो वाई कांट वी फॉर्म ए डोजेज फॉर्म की जिससे हमारी कम्युनिटी को हेल्प मिल जाए उस पर्टिकुलर मसाला से. सो दैट्स आवर मोटो टुडे. So then in 1920-30s, vitamins, vaccines were introduced. Then if we move to the 1940s, antibiotics era was there. R&D boost due to world drug discovery. So in 70, the rise of biotechnology was there, use of IT, how the IT could be used for biotechnology purpose, or the combination of IT, pharmacy. टुडे यू आर फाइंडिंग कि कोई लाइफ नहीं है आपकी कंप्यूटर के बिना. इंफॉर्मेशन टेक्नोलॉजी, एवरी फील्ड इज़ रिक्वायर IT. सो वी हैव टू, ये नहीं है कि हम फार्मेसी में हमारा सब्जेक्ट नहीं है कंप्यूटर का तो हमें नहीं पढ़ना है, नॉट लाइक दैट, बट वी हैव टू इम्प्रूव अवर स्किल्स ऑल्सो इन दैट वे इफ़ वी वॉन्ट अवर द मार्केट वैल्यू शुड बी मोर. तो इफ़ यू विल बी हैविंग द कॉम्बिनेशन, डेफिनेटली नो इंडस्ट्री विल बी देयर कि जो आपको प्लेसमेंट के लिए आपकी डिमांड बहुत रहेगी. यूर डिमांड विल बी टू मच एंड यू विल बी हाईली प्लेस्ड, और आपको पैसा भी ज़्यादा मिलेगा. व्हाट वी वांट, एवरीबडी वांट्स मनी. तो वो तो एक है, डिज़ायर है, दैट्स वी आर नॉट डिनाइंग, बट आप उसको अच्छे वे से ले सकते हैं उस पैसे को, इफ़ यू विल बी इम्प्रूविंग योर स्किल्स.
So then we are having 1960s, 50s में आपको new technology, discover of DNA, then breakthrough of etology, 60s, 70s, then we have moved to 80s, 90s. So this is how we have and you are finding in 2020 to 2020-21 में हमारे पास कोविड वैक्सीन्स आए ना, which has not been mentioned because these are the newly added things. So how they've been generated in a very short period, although the crisis was there, people were after these कि इसके clinical trial नहीं हुए, इसको proper way से नहीं, that's the from the political point of view. If we just think from the pharmacy point of view, really, I mean, our government has done wonder how the people been saved from COVID. Otherwise, crisis would have been there and many deaths would have been occurred. And our government has really prevented those deaths. And today you are finding that we are not that scared. Even we are vaccinated. अगर किसी को होता भी तो हम कहते हैं लाइट वे से फीवर होगा, आपका 3 दिन में ठीक हो जाएगा. अगर आपको टेस्ट नहीं भी करवाना, यू टेक दिस डोलो पैरासीटामोल खा के और अपना रेस्ट करिए, डाइट लीजिए, और यू विल बी फाइंडिंग कि 3 days में ही दैट रिकवरी इज देयर. सो दैट्स वंडर बिकॉज it effect is because of the people being vaccinated. So that's why we are safe and we are feeling happy over here. Otherwise, we would have been crisis in this time. So this is who has done this? Our technology, our pharmacist, our young generation been involved. And of course, the seniors' advice was there, but people who is the working hand is always the young generation. So we can give the advice कि आप ऐसे ऐसे कर लो, but after all young generation ही होगी जिसको काम करना है ना. So that's what good thing is there कि in 2021 में we have launched the product of vaccines for COVID. So that's the achievement of India and how the people been saved. Then the pharmaceutical R&D, we can say, in which you had a discovery, and from the discovery point of view, you will be finding that we will be having research born, then how the nano formulation development is there, and then it's been given to the human beings for gamma scintigraphy studies. And through gamma scintigraphy studies, we will come to know that how the distribution of the drug is there. That is related to your ADME. So it doesn't mean that we are pharmaceutics people or we are from pharmacognosy background or we are from which background, doesn't matter. But we have to cover these factors. Pharmacy is pharmacy. Of course, I mean for seats distribution, from PCI point of view, from council's point of view, the subjects been divided. But we know even one subject, we don't aware of the one subject, we cannot move ahead. For example, even today you are synthesizing so many molecules, and I'm wondering, people are saying we have synthesized 300 molecules, you know, in our organic chemistry. But where is that 300? Even in our department it was there, in other department it was there, and so many times I have requested them, please give me one compound. Please give me one compound. Then they were saying कि we have synthesized in such a way that we could able to characterize it. NMR करा लिया, IR करा लिया, mass करा लिया, बस उतना ही था हमारे पास. Otherwise, then they will shout कि हमने इतने compound बना लिए, अपने इतने compound बना लिए. But where are those? So why can't we think in that way कि हम 3 में से उसको screen करके, कैसे screen करेंगे? Through drug discovery. क्योंकि animals में हम पहले नहीं चले जाएंगे. We don't want to kill those, I mean the pretty animals. तो उससे हमें क्या करना है? तो we have to go for in silico studies. Of course, in silico studies also not that reliable कि we can rely on that studies, but at least we can get the idea. And from that idea, if you will move for the animal studies, that is always better. What people are doing, 300 कंपाउंड किए, अपने आप से उसको स्क्रीन कर लिया, उसमें से 5 निकाल ले, उसको एनिमल्स को दिया, 15 मिनट में एनिमल्स मर गया, बस हमारी तो काम हो गया. वी हैव राइटन ए थीसिस, हमारा पेपर भी आ गया. हम शो ही नहीं करेंगे हमारा एनिमल मरा ना, वेयर वी आर शोइंग इट. हमारी एक्टिविटी आ गई, हमारा एंटी इन्फ्लेमेटरी एक्टिविटी आ गई. वी डोंट बॉदर अबाउट द टॉक्सिसिटी स्टडीज़. हमारी एक्टिविटी आ गई, अब हमें थीसिस लिखना है, अब हमें प्रोजेक्ट्स डाल देना है. दिस थिंग शुड नॉट बी देयर. कभी कभी करना पड़ता है बिकॉज ऑफ टाइम शॉर्टेज की वजह से, या टीचर्स लोग बिजी ज़्यादा होते हैं, तो कभी थोड़ा बहुत मैनिपुलेशन करना पड़ता है, बट नॉट लाइक कि हम कहते हैं हमारी एक्टिविटी आ गई, हमने इतने कंपाउंड्स बना लिए, देन वी आर हैविंग दीज मैनी पेपर्स कि हमारे इम्पैक्ट फैक्टर में आ गए, हमारे थॉमसन रूटर इम्पैक्ट फैक्टर में आ गए. बट वी शुड बी ऑनेस्ट. दैट्स अगेन माई एडवाइस. सो इन द स्पैन ऑफ 10 to 12 years, what we have done is, that is we have to go for the, even for the nano formulation, as we know that they are having too much toxicity also. The reason I will tell you why they are having toxicity. Just say for example कि we are having 2 chapatis. One is made from atta and one is, that's wheat, that's what we are eating, and one is made from maida ना. तो कभी भी आपकी मदर्स या आपकी दादी या आपके घर वाले कभी एडवाइस नहीं करेंगे कि आप मैदा की रोटी खाइए, जब दोनों ही वीट है. तो क्यों नहीं खाइए, आटे की क्यों खाइए. सो दैट विल बी हैविंग द मोर फाइबर्स, डाइजेस्टेबल है. वो जो आपकी नैनो साइजिंग हो जाती है मैदा में, वो आंतों पे चिपक जाती है. आप डेली मैदा खाएंगे तो बीमार पड़ जाएंगे, बिकॉज योर इंटेस्टाइन विल बी लाइंड बाय दोज मैदास मटेरियल, एंड अल्टीमेटली जो इंटेस्टाइन की विथ है आपकी स्मॉल हो जाएगी, उसमें से फूड जा नहीं पाएगा. तो टॉक्सिसिटी इज देयर, बट वी शुड बॉदर अबाउट द टॉक्सिसिटी अलॉन्ग विद द एप्लीकेबिलिटी ऑल्सो. इट्स नॉट ऑलवेज एवरी पैरासीटामोल भी टॉक्सिक है, एस्प्रिन भी अगर आप ओवरडोज ले लेते हैं तो वो भी टॉक्सिक है. तो एवरीथिंग इज टॉक्सिक, बट वी हैव टू मेड आवर लिमिटेशंस, व्हाट वे वी हैव टू गो. सो दैट्स व्हाट, इफ यू आर formulating nano formulation, so first thing is we have to see the ADME, और कितनी हमारी ड्रग जा रही है, कितनी उसकी कॉस्ट पड़ रही है. ये नहीं है कि वी आर फॉर्मुलेटिंग नैनो फॉर्मुलेशन बट द कॉस्ट इज़ 5 times हाई. तो नो इंडस्ट्री विल लाइक टू हैव, एंड नो पेशेंट्स भी, वी हैव टू सी द पेशेंट्स कि वो अफोर्ड कर पाते हैं या नहीं. सिर्फ हमने बना दी उससे काम नहीं चलेगा, बट वी हैव टू सी कि उसकी अफोर्डेबिलिटी क्या है, उसकी इंडस्ट्री एप्लीकेशन, कोई इंडस्ट्री हमारा प्रोडक्ट लेना पसंद करेगी या नहीं करेगी. वी हैव टू थिंक इन दैट वे. सो दिस इज़ फॉर ऑल फॉर द यंग जनरेशन, जो बहुत जल्दी होती है हमको कि हमको आगे बढ़ना है, आगे बढ़ना है. तो आगे बढ़ सकते हैं, बट यू हैव टू पिक अप द थिंग्स एंड यू हैव टू गो फॉर द प्रॉपर वे. आपको प्रॉपर रिसर्च करनी है, कोई भी स्टेप छोड़ना नहीं है. अगर आपने एक भी स्टेप को ओवरटेक किया या उसमें शॉर्टकट मारा तो नहीं चलेगा. ओके, सो अर्लियर यू नो, फॉर लॉन्चिंग ए वन ड्रग, फुल डोज फॉर्म बनने के लिए आपने भी सुना होगा, पढ़ा होगा, इट वॉज टेकिंग समवेयर 25-20 years टू टेक वन डोजेस फॉर्म, to bring one dosage form into the market. But now you are finding the period has been reduced. How it has been reduced? From drug discovery only, from artificial intelligence only. So that's the, the modern techniques we have to adopt. So this is the drug discovery. Then we have to go for the development. After development, we have to come for the FDA. Covering all the points, like the toxicity studies, then GLPs in the GLP labs, then we can move to the FDA for the approval purpose. So we are having the target selection, then that is from the cellular and genetic targets. We are having genomes, proteins, bioinformatics, So today, I mean, many students are opting from bioinformatics. No wonder it is very, I mean, it's good thing. We have to see that how we can explore ourselves in a different fields, not limited to M-Form. हमने M-Form Sutics में कर लिया M-Form, but we have to gain the proper knowledge. If with M-Form you are doing some kind of the bioinformatics studies, then we are having the statistics studies, तो आप देखिए आपके मार्केट में कितनी वैल्यू हो जाएगी. So with little effort you can gain too much things. So when we are having the lead discovery, medicinal chemistry, then we are having the in vitro studies, in vivo studies, clinical trials, then afterwards we can go for the filing the new drug application to the DCGI office. If it is from the herbal moiety, then we have to move for the Ayush. So as we know, the Ayush is a combination of what? Ayurveda, Yoga, Unani, Siddha and homeopathy. So wherever we are finding our strength is more, accordingly we have to move that council for the filing the application, and then they will be licensing you, and very well you can go for the manufacturing of that particular drug. If it is from the synthetic source, so then little we can say process is lengthy. We have to go for the clinical phase studies, and after clinical phase studies only when we can go for the filing of the application. Even many industries are coming up nowadays also from the herbal sources also. Even you will be finding in our Indian pharmacopoeia many of the herbal monographs have been added, and our Jamia Hamdard team, including your Madam Dr. Vidhu, who is sitting, and next lecture you will be having from her, and she is working for the phytomedicines, and she is in the DCGI office committee also, where the selection for particular drug, how it will be managed, whether we have to give license or those or not. Okay, because there is a stringent requirement of that also, and so for not many industries are coming up from the herbal moiety, because expenditures involvement is there, and every industry want those reference standards could be made from 4 to presently we require 4 reference standards, and for industrial point of view they are very costly. Okay, even you know one reference standard costing 10 milligram is 40,000, for example, lycopene. So for getting those reference standards is really costly affair for them, and they want, industry want that they could be reduced from 4 to 2. But so far our office is not, drug office is not agreeing, and that's why many industries have not come up yet. They are trying, okay, but maybe very soon they will come up with the herbal drugs also, which are included in our monograph of IP. And if you will find 2022 monograph, around more than 100, I don't know the exact number, but more than 100 monographs from the herbal drugs being added over there. Okay. So this is the discovery research. Discovery research we can take to the innovative part. And again, all the process we have to cover, like target selection, lead discovery, medicinal chemistry, in vitro studies, all these parameters we have to cover for that reason. So, sorry, so we are having the discovery research, innovative part. Then we can make the compound libraries. How you can make the compound libraries? For example, you are carrying SPL studies, or you are carrying HPTLC studies. So every time getting reference is very difficult, or biomarkers which we call it as. So every time getting those biomarkers and bioactive is very difficult. Difficult means it's expensive affair. So what industries are doing, and what our analytic division is doing, they are restoring that data in the library. So they are making in HPLC kind of the library, that whenever you are doing the next time analysis of that particular sample, you can use that library. You need not to use that particular reference standard again, because you have developed those methods and now you are the own developed methods. So you need not to have the reference standard if you are storing the data and if you are making the library. So this is what we call as the library, means storing the particular data in your own system. For example, you are having HPLC system, you are having HPTLC system, whatever data you are generating, you just store it. Okay, so that's your own data. So every time for that particular drug, you can use that data. So what are the risks in research and development of pharmaceutical drugs? Why our samples are failing? You know, what's the reason? Because every time we cannot be have the success hand with us. So there will be the failures also. Whenever you are optimizing the formulations, there are always failures. You know, one formulation, that's the curcumin which I was mentioning to you, it took us 2 years to optimize it. कभी उसमें स्टेबिलिटी डाटा गलत आ जाते हैं, कभी सॉलिबिलिटी डाटा गलत आ जाते हैं. देन वी हैव टू डेवलप आवर ओन मेथड्स, हाउ वी कैन stabilize our preparation, how we can optimize. And the people who are really working in the R&D industries and in bulk drug division, they might be knowing better than me, because I've never worked in industry, although I know that these steps have to be taken, and I keep on discussing with drug office, keep on discussing with the industry people, and that's why I always been excited to talk with them. DCGI में सब जानेंगे कि हाँ, she talks about the drug discovery, she talks about the drug formulation, and sometimes they ask me कि मैडम आप कौन सी इंडस्ट्री में काम करते हैं. I said no, I've never worked in industry, but I keep on generating the knowledge कि मुझे, क्योंकि मेरे को एक ही I'm in the life शौक है कि मुझे फॉर्मुलेशंस लानी है मार्केट में. सो दैट्स माई पैशन, और वो भी हर्बल लेके आनी है. So that's why I keep on discussing, keep on asking from DCGI office and keep on fighting कि आप 4 reference में से 2 क्यों नहीं ले लेते, ताकि हमारे पास ज़्यादा clients आएंगे. We will be having the more manufacturers with us, we will be having the more products out of it. But अभी, I mean, it's going on. The day will come when they will reduce it. But so far it is 4 reference we require. So the reasons for failure includes lack of bioavailability, because we are not going the proper way, ADME, proper way, that what is the bioavailability of the drug, because it requires little time. First off, you will be going for the formulation, and after that you will be requiring absorption, where it will be absorbed, how much it will be absorbed, then all the proper ADME, it requires little time. So maybe 5 to 6 years to recover these. But we are always in hurry कि हमारा जल्दी जल्दी सब हो जाए, research भी हो जाए, product भी आ जाए, industry से भी बात हो जाए, always. So, but it requires little patience. With patience you will achieve it. ऐसा नहीं है, always achieve it. So there's the lack of bioavailability, toxicity, then lack of efficacy, but also पेटेंट पोजीशन, मार्केट प्रपोज कंपीटिटर्स. कितने कंपीटिटर्स हैं आपके प्रोडक्ट के मार्केट में? वेदर यू विल बी फॉर्मुलेटिंग, व्हाट विल बी द कॉस्ट ऑफ इट, व्हाट्स द ऑलरेडी रनिंग कॉस्ट ऑफ द प्रोडक्ट, वेदर योर प्रोडक्ट विल बी competing with that marketed product or not, because many companies are there from years together they've been doing. So you will be the new player in the market. So whether you can compete with those multinational companies, or your product will compete with those multinational, that's the big question mark. So that is where our products failed. No, अगर हमारी cost ज़्यादा पड़ गई, because our is the new product, people don't know my product, ना. If I'll go today in the one product in the market, so who say who is this company, which is this company, we never heard about this company. We will always having like the product of Cipla, Sunpharma, Dr. Reddy. Why we should have the product of this company when the cost is, even my product is costing me more? because they are manufacturing in bulk. They are not doing now the research. I have to do the research, optimization. They have already done those process. So their product is always in the market. तो मेरा product तो महंगा भी बनेगा और कोई मुझे जानता भी नहीं है. Then that's the work of the business development. Who will be developing your business? How you can create the business? Then what is the value of your product? बनाने को तो we can make today 10 product. But where is the marketing of our product? That's the big question mark. So we have to build our marketing. How we can build our marketing? Just creating the new ideas. आप उसमें कुछ नया essence डालेंगे. If you will not put any essence to it, nobody. जैसे for example food ventures भी हैं. You will be eating से आप McDonald का वो खाते हैं burger ना. Tomorrow some other company has come up. Why you will have the other company's burger when you having the बर्गर्स? आपको मैकडोनल्ड का पता है स्टैंडर्ड कंपनी है, एमएनसी है, इसका तो अच्छा होगा, हाइजीनिक वे से बना होगा, मोर वैल्यू इज़ देयर, मोर दैट कंजम्पशन इज़ देयर, सब कुछ ठीक है, तो हम दूसरा बर्गर क्यों खाएंगे? अनलेस अनटिल वो चीप नहीं होगा या उसमें कुछ नया नहीं होगा, कुछ न्यूट्रिशनल वैल्यू नहीं होगी. वाई विल हैव दैट बर्गर? सो दिस इज़ द सेम वे. So that's where our pharmacist has to work very, very hard to bring those product in the market. So I like कि आप अपने यूनिवर्सिटी से एक प्रोडक्ट बनाएं और आपके MBA के स्टूडेंट्स उसको सेल करें. तो you will be having the interaction also, you will be having your own product also. That's one of the suggestion. Something went wrong. So we are having the therapeutic innovations, many different ways to innovate, including newer, better uses of existing molecules, treatments. So existing compounds, we can have the library screening and compound mining, new compounds through medicinal chemistry, new uses of existing compounds, new indications of existing drugs, improved formulations of existing drugs. Then we are having the completing registration, dossier. Many people are involved in the process of making the dossier, and some students, those who are having the innovative ideas, like people who are doing the research work and they've been collaborating with industry and they are preparing the dossiers for them, and making dossiers, they are charging quite heavy. Okay, एक scholarship भी मिलता है और एक dossier भी बनाते हैं. Their earning is better than a teacher if you are having the innovations in your mind. Okay, so jitna, the teachers are getting, more than that they are earning, and they are scholar also, and they are doing kind of the innovation. This is what innovation is there. So then we can divide this process into discovery, preclinical, clinical, and access to patient. So drug discovery methods, we are having the random screening. आप स्क्रीन कर लेते हैं. फॉर एग्जाम्पल, एज आई गिवन यू द एग्जाम्पल कि आप ड्रग डिस्कवरी से आपने 300 कंपाउंड्स बनाए, उसमें से आप स्क्रीन करेंगे रैंडम बाय यूजिंग योर माइंड कि आप, बिकॉज यू आर हैविंग योर ओन माइंड, एवरीबडी इज़ हैविंग योर ओन ब्रेन, ओन माइंड. आपका माइंड किस तरफ चला जाता है, वी नेवर नो कि आपको क्या करना है. दैट्स यू नो कि मुझे क्या करना है मुझे, तो उसमें से स्क्रीन कर सकते हैं हम. तो दैट्स द फर्स्ट स्टेप. वी आर हैविंग द रैंडम स्क्रीनिंग कि इफ़ वी आर हैविंग वेरी गुड केमिस्ट्री स्ट्रक्चर कि वी वांट टू नो कि अगर हम OH को रिप्लेस कर देते हैं COH से, तो मे बी आवर सॉल्युबिलिटी प्रोफाइल विल बी इम्प्रूव्ड, मे बी हमारी फार्माकोलॉजिकल एक्टिविटी विल बी इम्प्रूव्ड. सो दैट्स व्हाट वी हैव थिंक, दैट्स what in silico studies will suggest us. So we have to go for, that's your the artificial intelligence in that way. So molecular manipulations means कि आपने OH group हटा के COH कर दिया, OH3 कर दिया. जैसे for example, today you will be having diclofenac for example. So in diclofenac, where we are having the external we are using diethanolamine. And whereas for internal, what we are using, anyone from the students? Diclofenac, what? Hydrochloride, citrate, sodium. So diethanolamine sodium is for internal use, and that's for external. When we are formulating the gel, like the various gels are available in the market, like Volani, Omani, and whatnot. Others are making controlled release gels also. Very soon you will be getting in the market. That's the triple strong action, double strong action. So because the company always thinks kind of the innovations, how people will be bored from one product, but the other innovative product is there. So they will be making the diclofenac sodium in double strength. And maybe very soon you will be having in the market this product, which will be having the double strength. Of today, 1.12% of the diclofenac is used in gel preparation. Whatever it is, Volani, Omani, and other makes are there. But very soon you will be having the double strength of the diclofenac diethanolamine. Reason is, they just think we will be having for the controlled release. According to me, which is not that much possible, because when I have done skin permeation studies, really I was not very satisfied. Maybe they will be having kind of the innovative ideas on which they will impress consumers, and they will be having the more sale of the product. So sometimes we as a scientist, we cannot think in that way what the business development team thinks. So that's why I always tell you कि आप MBA वालों से collaboration करके और कुछ product बना के उसको कुछ कीजिए, marketing करिए. So then molecular designing, molecular metabolites, serendipity, that's again from the old thinking. जैसे आप देखते हैं कि रामायण में वो हनुमान जी क्या लेके आए थे, संजीवनी. So what is संजीवनी? पेड़ ही तो था, plant है ना. So similar type of the plants, why can't we explore? कि हम जैसे जाते हैं जंगलों में, तो हमें किसी ने बताया है कि ये प्लांट, आप देखिए कि एक प्लांट है that is जो कि हमें इरिटेशन कॉज करता है. व्हाट इज़ दैट प्लांट? खुजली हो जाती है. दैट्स नॉट कमिंग इन माई माइंड. उसके साथ ही एक प्लांट और भी होता है विच वी आर नॉट अवेयर. समटाइम्स इफ़ यू मूव टू जंगल, तो कई प्लांट ऐसे हैं कि जब आप उनके पास से गुजरते हैं, इफ़ यू दे विल टच योर स्किन, तो आपको इरिटेशन, खुजली होने लगती है. बट पीपल हैव टोल्ड कि उसके साथ में ही एक प्लांट है, आप उसको रब कर दीजिए, आपकी इमीडिएटली ठीक हो जाएगी. सो दोज प्लांट्स कुड बी एक्सप्लोर्ड इन दैट वे कि उसमें ऐसा क्या है कि जिससे खुजली हुई थी, इरिटेशन हुई थी, और उसमें क्या है ऐसा कि जो इसने हमें ठीक किया. So in that way you can bring the combination, and that's one of the good, the innovative idea. And what is your the serendipity is here. तो अगर हम उनके पुरानी बातों को ही याद करें, रामायण की, जिसमें कि संजीवनी लेके उन्होंने लक्ष्मण जी को मूर्छित होने से बचाया, उनके लाइफ को सेव किया. सिमिलरली वाई कांट वी हैव दोज काइंड ऑफ द प्लांट्स. और अगर हम संजीवनी को ही लाएं और उसमें देखें कि क्या है ना, पीपल माइट हैव एक्सप्लोर्ड, मुझे नहीं मालूम अभी. सो टारगेट सिलेक्शन. सो दिस इज़ व्हाट टुडेज डिमांड इज़ देयर. टारगेट सिलेक्शन से क्या है कि आपको डिसीजन लेना है टू फोकस ऑन फाइंडिंग एन एजेंट विद द पर्टिकुलर बायोलॉजिकल एक्शन दैट इज एंटिसिपेटेड थ्रू हैव थेरापेटिक यूटिलिटी. फॉर एग्जाम्पल, people are suffering from colon cancer. So we are giving a drug which maybe some of the amount could be absorbed at stomach part or intestine part, and by the time it reaches to the colon, it may be having say from 20 mg, 10 mg ही बच गई है drug. So if you can save that drug, 20 milligram of the drug, and you want it should be released at colon only, so why can't we, those drugs could be explored and they can be coated with particular molecules, particular polymers, surfactants, and today if you will be finding the kind of the surfactants, many newer surfactants are coming up to the market which we can explore. And I'll tell you, the company like Gatifos is one of the company which is having the wonderful excipients. Of course, I am not promoting that company, but I am using those excipients, and I find it they are very good solubilizing agent. You will improve the quality of the solubility, and once we are improving the solubility, we all know the bioavailability parameters will be improved. So this is how we have to select, how we have to go, and can we have the combination of the surfactants by which we can achieve all our wanted parameters, what we want. So those parameters could be very well we can achieve by the combination of the surfactants, and today we are not stick to the one particular status कि हमें यही use करना है, हमें HPMC ही use करना है, हमें HPLC ही use करना है, ऐसा नहीं है. We can explore other excipients which are coming to the similarly, not from the pharmaceuticals point of view, but from the cosmeceuticals point of view. In cosmeceuticals, many of the product, for example, you are having anti-wrinkle product, anti-acne product. We want, even if the drug will not reach to the blood, at least it reaches to the deeper skin, like as you are finding, the epidermis is having 5 layers, and the stratum corneum is the main hurdle for us. Why can't our drug could be penetrate through stratum corneum and could reach to the up to the at least deeper to the skin, not to the blood I am saying, because we don't want that our excipient should reach to the API should reach to the bloodstream sometimes if the drug is not for internal use. So we want it should stick, it should reach to the deeper. Why can't we have the good penetration enhancer through which we can improve the penetration and our drug reaches to the proper system? Like in the case of the anti-acne, if you will be having the kind of the good formulation for anti-acne, you need not to taken the internal formulation sometime. And similarly, we talk about the anti-wrinkle. Of course, anti-wrinkle we always talk about the fashion, but it's not for the fashion purpose. The drugs which are having the phytomedicines which are having the good antioxidant properties, like I had given you the example of the hemp seed oil earlier. So that can be used for anti-wrinkle purpose. I mean, everybody wants to have the beauty. I mean, whosoever is there, कोई भी नहीं कहता कि हम जल्दी से हमारी झुर्रियाँ पड़े, wrinkles आए. Everybody wants to maintain. So why can't we have these simple, cheap things, and we can improve the skin profile? So this is what, again, the target selection, and target selection is depend upon us where we are going, and the target validation to prove that manipulating the molecular target can provide therapeutic benefits for patient. So that we can again have the cellular or genetic targets, genomics, proteomics, bioinformatics, target selection, lead discovery, medicinal chemistry, in vitro, in vivo, clinical trials. आप लोग बोर तो नहीं हुए गए? Because I am finding कि कुछ मैं अपना lecture stop कर सकती हूँ. If you are finding बोर तो मैं I can stop over here. Time क्या हो गया? बोर तो नहीं कर रही? So ये तो हो गया आपका. So target selection, for target selection we can go for the biochemical classes of targets, G protein couple receptors, which is the demand of today's, enzyme selection, hormones and factors, ion channels, nuclear receptors. So techniques for target identification which can be used again, that's again we are having the conventional system, target selection, lead discovery, medicinal chemistry, in vitro, in vivo and clinical trials. So cellular and genetic targets which involves the identification of the function of a potential therapeutic drug targets and its role in the disease process. For example, we are having the small drugs that involves identification to the receptor, to the targeted, and you might be heard about the one drug, that one drug has come up in the market, not in Indian market, but in the USA, which has improved the health management, and also the patients, cancer patients have been recovered from various kinds of the cancer by taking that drug. That molecule is not coming in my mind, but that is the antibodies preparation which been conjugated. So now you are finding many protein and peptides are coming up into the market, and these protein and peptides are acceptable over to our system. So that's why if we look for although the problem with the protein and peptides is the solubility problem, bioavailability problem, so for that we are here only. We can improve those factors, and very well those products could be at least launch नहीं कर सकते, but हम अपना काम तो कर सकते हैं कि research का हम उस level पे कर दें कि many other companies can take up those who are having having the finances problem. Okay, we cannot have those finances. But those companies, multinationals, they can take up your molecules at the initial level, and they will accept it. So that's what cellular and genetic targets is there. Then we are having the genomes, the study of genes and their functions. So what the genomics aim to understand the structure of the genomes, including the mapping, and you are finding for example, the Biocon company is there. So that company is working on this principle only. They are just mapping genes and sequences the DNA, and very soon they may come up with the molecules. They are under clinical trials, some of them under clinical trials, and some of them are under optimization, and maybe in coming 4-5 years you will be having the many wonder molecules from the Biocon which will be based on this genomics. The problem with the genomics is when we are going from batch to batch optimization, many problems are coming up. I mean, they are not as similar as the first batch was there. So many variations are there. So that's where we need the optimization. It's not the simple molecule, your API, simple drug molecule, where the optimization is very easy. But here the optimization is very difficult because from batch to batch, many variations are coming up. So that's what we have to see. So this is genomics. Then we are having the proteomics. Similar way, we have to see the protein scale separation and identification, then bring to the dosage form. Again the proteomics, then bioinformatics field, which I was mentioning you, with the application of the bioinformatics, you will be having the target identification, computer screening of chemical compounds, and pharmacogenomics. Now the pharmacogenomics and pharmacovigilance, they are coming up the fields, and as you know, Indian Pharmacopoeia Commission has established a high-level committee for this reason, and they are coming up with the pharmacovigilance program in collaboration with WHO. So then lead discovery, what is it? Identification of small molecules, modulators of protein function, then again transforming to the drug synthesis and combination. Then we are having the combinatorial chemistry. That's the rapid synthesis of a computer. That's what we are talking about, the drug discovery and the use of your computer systems, where we are having the ADM properties, reduce toxicity and eliminate side effects. Then the assay development is there. Then high throughput screening of, that's what I have already given you the hint also. Then high throughput screening, these are the, we can say you are the high tax machineries which are used for the high throughput screening. That's for the drug discovery purpose. Sir, we are in the medicinal chemistry, we all know about where we drug discovery, lead optimization, process chemistry and development, library development, which we have already discussed, SARC study, that is a structure activity relationship we have to develop, and this again for the identification and for again we can say the screening of the active constituents, then SRS studies for one morphine molecule, then in silico screening based on the finding structures, and they are most likely to bind to a drug target. Then we are having the chemical synthesis, we all know about these in vitro studies. And of course, now the in vitro studies, what we were doing, the dissolution studies. Besides these dissolution studies, we are having the cell line studies. So many people think that the cell line study is the in vivo studies, but it's not that. It's only the in vitro data you are getting with the cell line studies. After cell line studies, it's giving us the hint that how much drug has to be used, what is the toxicity, but again we have to do the animal studies, then only we can, or the dynamic studies, then we can conclude our data, not give it the cell line studies only. So in vitro studies, then drug affinity and selectivity, again the binding with the which enzymes, receptors, transport systems. So selectivity, drugs should bind to specific receptor site on the cell. This is the example. So cell disease model, there are the different models are there. Now the stem cell disease models are going at a very high level, and kind of the clinical studies are going on for this. Then we are having the lead candidate refinement, where we see the potency, oral viability, selectivity, kinetics properties, safety, in vivo studies. So its experimentation using a whole living organisms gives information about metabolic profile, toxicology, drug interaction. Then we are having the animal models. We have to select the animal models according to what we want. We are having the SCID mice, NOD mice, Rero for the gene function. For the different functions, we will be having the different kinds of the mice or the animals could be used. Somewhere we are using the rabbits also. For example, if we have carried out the gamma scintigraphy study is also kind of the in vitro study, and the gamma scintigraphy study is always carried out in a big animal like rabbit. So behavioral studies, then we are having the functional imaging. That's with the gamma scintigraphy, we can all know about MRI, CT scan. Then ex vivo studies, what we are doing, kind of your ex vivo studies, what the skin permeation is studying is used, how much drug is penetrating, that's your only the ex vivo studies, where we are using the skin of the rats or the animals. So that's why it is called as the ex vivo. We are doing in kind of in vitro, but we are using the animal in some way or what way. So for example, for internal preparation, you are using the rat gut permeation. So through intestine, how much the permeation of the drug is there. So for internal, we are having the rat gut permeation. For external, we are having the skin permeation studies. So these studies we can plan. Then the clinical trials, of course, it can go up to 7 years. So please 0, recent designation also known as human microdosing studies. So these all studies, preclinical, pharmacodynamic, then we can show the efficacy of the product. Phase 1, आप सबको पता है phase 1 में how many volunteers are needed. Then phase 2 studies, how many volunteers, 50 to 300 patients, and here the volunteers are healthy in phase 1 and phase 2, whereas the phase 3 is the disease one. So phase T is extended clinical trials, most expensive and time consuming. We need 250 to 1000 patients, and we have to see with the safety, efficacy. For that reason, what the volunteers been selected, we have to give the kind of the insurances to them also. कुछ किसी की death हो जाती है तो वो क्या करेगा, उसकी family क्या करेगी. So now the DCGI has added this point also कि आप उसके लिए कुछ insurance के लिए पैसा रखिए, जो भी volunteer आता है study के लिए. Of course, what the remunerations we are paying to them. Besides this, we have to give the compensation also if the loss of the life is there. Sir, we are having the phase 4 studies, this we all know. So recent developments, we are having the nanotechnology, then your gene therapy, dual gene therapy, orphan drugs. So orphan drugs we all know, I mean that been developed specifically to treat a rare medical condition. So again, for these orphan drugs, many of the clinical trials and studies been going on. So rare studies, these are the where the rare and the orphan drugs are there. Then we are having the nano-cosmeceuticals. I'm not giving much time, sunscreening, skin. These are all the nano formulations, anti-wrinkle products which have been developed, some of the products by us also. Hair care, skin cleaners, they are all in the kind of the nano formulations are coming up into the market. Topical drug delivery system for phytochemicals. We are having the many phytomedicines which can be used for topical drug delivery system. Nerve degeneration, nano gels been used, and then cancer spit test. Then these are the, thank you for patience listening. And if anybody is having the kind of the question or discussion, we can have afterwards any kind of the discussion. So thank you for patience listening.
Thank you, ma'am, for guiding us on the different aspects related to evolution of drug discovery, its method, the benefit and risk ratios, and its approach to recent innovations, creating a huge difference in the pharma background as well as for the welfare of people. Thank you so much.
Thank you so much, ma'am. It was a Herculean task to take a review of drug discovery right from lead generation to formulation, and she did an excellent justice to it, as she had to cover it in an hour or so. Thank you so much. It was very enlightening. Just to summarize her talk, I am not reiterating what she said, but the message, the broader message which we should take from her talk is, we should stop compartmentalizing our faculties in pharma. Like I am from pharmaceutics, he is from pharmacognosy. So those compartments should start dissolving. Not only pharmacy, we should also integrate all other faculties like law and engineering and all, and then only we would be able to take our profession to a new height. So the world is moving towards interdisciplinary work, and we should also move ahead with it, otherwise हम वो जो stream है उससे बाहर हो जाएंगे. Just to lighten the academic atmosphere, I would like to give you a small example which is not at all academic, की कसं सगळं एकाच दिशेने जात आहे आणि आपण जे म्हणतो की आमचं फार्मसी, आमचं फार्मास्युटिक्स, आमचं फार्माकॉग्नॉसी, आमचं इंजिनिअरिंग, तसं काहीच राहिलेलं नाही आहे. ह्याचं एक छोटं एक्झाम्पल देते आणि थांबते. आय एम लर्निंग फारसी these days because I am very interested in Farsi and Urdu. So when I started learning Farsi and Urdu, I realized that 65% of the words we use in Marathi have come from Farsi. आणि मग माझा जो भ्रम होता की आपली मराठी, प्युरेस्ट मराठी अशीच बोलली पाहिजे, तो सगळा निघून गेला. एक साधं एक्झाम्पल असं आहे की आपण अफलातून हा शब्द नेहमी वापरतो बोलताना. फ्रॉम वेअर दिस वर्ड हॅज कम, but what is the etymology of that word? We have heard about Aristotle and Plato, who were the great philosophists, right? So Plato, when it went to Arabs, they can't say P, so they pronounce P as fa, and they have a habit of attaching a before. So they started calling Plato Aflatu, and they started calling Aristotle Arastu. Okay, and because Plato believed in a third world, a world of forms which exist above this materialistic world, when we say Aflatun, it is something which is out of this world. Okay, and whenever in Marathi we say Aflatun, that is a word which has come from Farsi, and we are actually taking name of Plato, who was a great philosophist. So then where are the borders? Okay, it's all merging, and so should we do when we are pursuing our faculties. So thank you so much, and let us strive our best to dissolve these borders and get aligned in development of new drugs and take our profession to new heights. Thank you so much.
Thank you, ma'am. Now, I would like to request Dr. Mrudula Belle ma'am to present the Dr. Kanchan Kohli ma'am with a certificate and memento as the token of gratitude. Please, ma'am. Thank you, ma'am. Now I would like to request Dr. Kanchan Kohli, ma'am, to present the Dr. Mrudula Bele, ma'am, with a certificate and memento as the token of gratitude. Please, ma'am. Thank you, ma'am. I request to please be seated as we start with our second session. I request Dr. Vidu Eri ma'am and Dr. R.A. Patil ma'am to please occupy the dais. The session is entitled as Phytopharmaceuticals, a holistic approach to plant-based drugs. For this session, we have with us Dr. Vidu Eri ma'am as a resource person and Dr. R.A. Patil ma'am from Kekewa College of Pharmacy, Nasik as the session chair.
I request Mrs. Shirke ma'am to please introduce Dr. R.A. Patil ma'am. Please, ma'am.
Good afternoon all, myself Mrs. Shirke, and I am very glad to introduce the chairperson of a second session, Dr. Rupali Patil, Professor, Department of Pharmacology at Kekewa College of Pharmacy, Nasik. Madam had completed her B.Pharm and M.Pharm from NDMUP College of Pharmacy, Nasik, and her PhD from JNTU in 2014. Madam have total 19 years experience in academics and 2 years of experience in industry. Madam have total 49 publications in national and international journals, and she had 3 books in her credits. She had a total citations 411 having H index of 10 and I-10 index of 10. She received 3 awards, one for research paper and another awards are for appreciation in her generous contribution in development of a college from Gokhale Education Society. Hearty welcome you, ma'am.
Thank you, ma'am. Now, I would like to request Dr. Vidu Eri ma'am to felicitate session chair Dr. R.A. Patil ma'am with bouquet, shawl and token of gratitude. Please, ma'am. Now, I would like to request Dr. R.A. Patil ma'am to felicitate Dr. Vidu Eri ma'am with bouquet, shawl and token of gratitude. Please, ma'am.
Thank you, ma'am. I request Dr. R.A. Patil ma'am to kindly introduce Dr. Vidu Eri ma'am. Please ma'am.
Thank you, Rucha. Today I am here to introduce our second speaker of the second session, Dr. Vidu Eri ma'am. She is Professor and Dean, School of Pharmaceutical Research at Jamia Hamdard, New Delhi. Madam did her Bachelor in Life Sciences from Delhi University, B.Pharm from Delhi University, M.Pharm in Pharmacognosy and Phytochemistry from again Delhi University, and PhD from Jamia Hamdard. Madam is Dean at SPER. She was head Department of Pharmacognosy and Phytochemistry at Jamia Hamdard. She also was production manager at Baxin Homeo Pharmacy Private Limited, New Delhi for 2 years, and she served Jamia Hamdard for 13 years at UG, PG and PhD level. As far as research profile is concerned, Madam guided almost 63 students for post-graduations, 11 students for doctoral studies. Madam is having 216 publications to her credit with a citation of 1655 as of today, with H index of 20, INT index of 35. 2 patents are there with her credit. Madam is life member of so many associations, to cite a few: Indian Pharmaceutical Association, Indian Society of Pharmacognosy, Indian Pharmacy Graduate Association, Indian Hospital Pharmacist Association, ISTE, APTI, and Indian Association of Analytical Chemists. With this brief introduction, I welcome you, ma'am. With the top, Madam is going to deliver a talk on phytopharmaceuticals, a holistic approach to plant-based drug session. Please, ma'am.
Thank you so much. Yeah, I'll just make it slideshow. Yeah. 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 Yes, so a very good afternoon to all of you. And right at the onset, I would like to thank the organizers for providing me the opportunity to interact with the learned audience. And I'm very thankful to the students who have been really patient, since morning and listening to us. So thank you so much. And you have to again sit for at least 1 hour. So I will make it little interactive, okay? Now, since being a professor of pharmacognosy and phytochemistry and taking up this topic of phytopharmaceuticals, because what is your topic? That is recent innovations, a holistic and integrated approach to pharmaceutical science. So, according to new education policy, we are always talking of integration, isn't it? And if you look at pharmacy as a whole, I think you must be UG students or PG students, maybe few of you are PhD students. So if you understand this, pharmacy is already an interdisciplinary science. Okay, so I think your profession is one which you are already interdisciplinary. So how you are interdisciplinary? There are 4 main departments and there are now 8 to 9 specializations all together. So what we have to do is we have to come together. All my senior speakers, they have spoken about this, and that is why you can understand that how we can take these herbals to a new, you can say to a new height, and sky is the limit. So you must have been studying pharmacognosy probably according to your new PCI syllabus, starting from 3rd, 4th, and then of course I think 5th, probably 7th. So it has been reduced, The subject of pharmacognosy, it has been reduced. But if you go in for masters, it is now only masters in pharmacognosy. But you have to understand that phytochemistry is a very important part which is associated with pharmacognosy. So when we talk about this phytochemistry, which chemistry is associated with plants? Anybody? Which chemistry is associated with plants? Huh? Organic chemistry. Your plant chemistry is organic chemistry, medicinal chemistry, natural chemistry, isn't it? So all things are already integrated. And which is the best method of classifying the crude herbs? See, we cannot now use the term as drug. You cannot use the term drug, so we use it as botanicals in pharmacognosy. Huh? So which classification is the best? Classification again depends on what you want to do with the plants. Okay? Now what are the offshoots of pharmacognosy and phytochemistry? Cosmeceuticals, nutraceuticals, supplements, all these are what? Offshoots. So it means that is why you say what? That it is the mother of all sciences. So probably at UG level when your teacher is teaching you, you think what it is. What we are studying the source and then different names and all these things, and you feel that, oh, you know, we are just cramming it, you know. ज़्यादातर students क्या बोलते हैं कि हम कुछ नहीं है, हम सिर्फ उसको रट रहे हैं. It is not like that. See, if you integrate, see here the term comes integration. That how you have to integrate your pharmacognosy with all the subjects which are there. Right? And you know, it is so difficult to work on plants. If a chemist is working on plants, he will just take out a single molecule, an isolated one, and work on it. And before that, what a pharmacognosist and phytochemist is doing, he's searching for the right identity of the plant. Isn't it? First, we have to give a right identity to the plant. And that is why the other definition of pharmacognosy is what? Study of poisonous herbs. Now, why poisonous herbs? Because if there is any substitution, yes, we can have it, but if there is any adulteration, so it will lead to poison, isn't it? So the first thing is that we need to give a correct identity to the plant. Now, how to give correct identity to the plant? You are using a plant not because it is just natural in origin. Why you are using it? Because there is certain chemistry in it, isn't it? There is a certain chemistry in it, and this chemistry, these phytoconstituents, you have to do what? You have to extract them. And before extraction, you have to do what? Processing. So identity, authenticity, and then extraction. So when you are extracting something, you need to know all the unit operations. Now which technique you are going to deal with, isn't it? Which technique you are going to take, and then which solvent you want to use. So which solvent you want to use, it depends again on what? The nature of the phytoconstituents. So you have to understand what? Polarity, the concept of polarity, isn't it? So once you extract it, then next what you do? You do separation, isn't it? You do separation. So when you do separation, what you are using? All the chromatographic techniques. So it is what now? Analytical chemistry is also associated with it, isn't it? So then what you do? You isolate it, isn't it? After isolation, what you are doing? You are characterizing it. So this is all about pharmacognosy and phytochemistry, and that is why, you know, there is a concept that how we can consider or how we can give a new concept to these extracts which are already there in our traditional systems of medicine. Okay, so now we are not saying that the traditional systems of medicine, normally people say that there is no science behind it. There is a very important science behind all these traditional plants. You have to give a substantial data. So what now we are going to do in phytopharmaceuticals is to give a data, a robust scientific data. So from where this robust scientific data will come? Starting from the extraction, phytoconstituents, and looking in for the 4 phytoconstituents. So already you have heard that we should know these marker compounds, and how to then isolate these marker compounds. And then finally what you have to go into? You have to go in for what ma'am was telling you about in silico studies, and then ex vivo, in vitro, toxicity studies, all this, so that we can have a new class of drugs. Okay, so now so let's talk about traditional medicines, which is the interface. So always remember that your traditional systems are the interface. And all of you know that India is a rich heritage, so already there are extracts which are well there in our samhitas. So now, what is the literal meaning of samhita? Pharmacopoeia. So how do you add a thing in pharmacopoeia? After doing lot of research. Otherwise you cannot put anything in pharmacopoeia. So it means that lot of research was put in, and that is why it was there in samhitas. It was not just like that somebody came and put all the medicinal plants. And moreover, again, nobody is questioning your traditional systems of medicine. What you have to tell them? You have to tell them the chemistry behind it, that which is called as natural product chemistry. Okay, so you have to be very good in natural product chemistry as well as this analytical chemistry. Because those who are doing masters, you know which is the common paper for all subjects? Which is the common paper for all subjects? Huh? Modern analytical techniques, isn't it? Huh? This is the common paper, isn't it? So why this is a common paper? Because the technique, the technology which goes on changing, it can be applicable to all these subjects. So what you have to learn? You have to learn the technology. You have to learn the technology, and you have to be well versed with the technology. And then only anything which is there, you can put it to a new height, right? So now this traditional medicine is the interface. Then of course I would just like to tell you briefly that what was the need for amendment, what was the thought process, what was the government initiative, and what was the stimuli to develop phytopharmaceutical. Why there was a need? We have so many pharmacopoeias are there, already formularies are there, so why we need to have phytopharmaceuticals? And then what is the constitution of these phytopharmaceuticals? And what is the way forward, and which initiatives you have to take. So suppose we understand all these facts, you can very well understand this. This is actually, if you develop a phytopharmaceutical, this will be an integrated approach. So you must choose, and I also urge the teachers here to choose the topic of the research very wisely. Just don't waste the time like that. If you choose something and take the help of at least 1 or 2 departments, and then further the same work can be given to the PhD work, then only we can come to something. Otherwise, you can go on, doing some activities or taking out the extracts, and then you take out the flavonoids. So if somebody asks you, okay, they say, these flavonoids are not acting. So how the flavonoids will act? You are heating at such a high temperature. Huh? You are doing oxalate extraction at a high temperature, and you say, oh, flavonoids are not working. So then you say, oh, I don't want to work on a medicinal plant because it doesn't work. Because you don't know that what is happening to the constituents. So you have to use the technique very wisely. So all this, when it is put into, you can come to a new class of drugs which are called as phytopharmaceuticals. Now suppose you are working on a traditional plant and making an extract, you cannot have your intellectual property right on that. But if you develop a phytopharmaceutical, you can have an intellectual property right. So we will just move and see. So you know that the Government of India published a draft, and so what was the main aim? That is to have a regulatory provisions of defining the phytopharmaceutical. So what it gives you? Now see, it is very important when I say 3 terms, that is quality, safety and efficacy. So see, we sometimes there can be little efficacy, but we cannot play with the safety of the patient. So that is why we have to go in for all other clinical trials, which will be also there. But how will you do, because a plant is having so many constituents in it. But at the same time, the government's initiative was, and the mind which was working behind was, that is, we have to keep intact the philosophy of our traditional systems or of the plants. That philosophy has to be keep intact. You're not playing with that philosophy. But yes, if you have to do a pharmacokinetic studies or pharmacodynamic studies, or you have to do toxicity studies, so at least you should know that what are those 4 phytoconstituents. So you can work on that, isn't it? Now, what happens is that sometimes they select the 4 phytoconstituents just like that, which are the common ones. Okay, so they say, okay, I have selected the phytoconstituent like quercetin or any other common one. So you know that these constituents or tannins, these flavonoids or quercetin or tannins, they are present in all the plants, because you know plant first protects itself. It is a living system, it cannot move, so it has to protect itself, and for protecting itself nature has given it what? Tannins, phenols and flavonoids, which will be there in all the plants. So it means that you have to be very specific when you are choosing those marker compounds. It has to be specific. But yes, the presence of these flavonoids and tannins is also important, because holistic approach which you are talking about and our traditional systems, that depend on what? The holistic approach. That is, you need to keep everything intact. But I will just come for this definitions. I'm not, I mean, you can just read all these things and I mark them, which are very important for the students. And if you want to know more about these phytopharmaceuticals, please visit the site of CDSCO. I hope all of you know what is CDSCO. Huh? So please visit that site, and there are many question and answers also given there, frequently asked questions, before you want to develop a phytopharmaceutical. You must be having many extracts, crude extracts. So you can develop a phytopharmaceutical. So this amendment, why the amendment was required? You know, there are 2 tablets which we all of us know that they are very important, very important. But you know how much time it took to come? Such a long time it has to come. That is more than a decade to get the approval. Now approval is a must. So it took almost a decade to come to the approval. So now what should we do? And moreover, all over the world, globalization, if you have seen United States pharmacopoeia or Chinese pharmacopoeia or Japanese pharmacopoeia, have you seen all these pharmacopoeias? Huh? So what is the difference in the monograph of the plant in all these pharmacopoeias? So please, I urge all the students to see all these pharmacopoeias and what is the difference, and then you will start seeing that, okay, even if you don't have a marker compound, you can develop a fingerprint profile. So in Japanese pharmacopoeia, which are very important extracts for them, what they have done is that they have developed the fingerprint profile. A fingerprint profile is developed, also telling you that in this, if it is an authentic herb, you will have 5 or 6 spots which are of this color. Just simply match this with the herb which you are getting. So this avoids all the confusion, and you can develop so many fingerprint profiles for all the plants. You can also check the adulteration. So this is a very good work, and you can also help to create a national wealth of some of the plants where the monographs are not there. This can come as a help from all from students who are working on it. Okay. So now if you see the international scenario, already they have done this. But their laws, the rules are not that stringent, which now we have developed. Because you know, every now and then they were coming with some herb or volatile oil, and they were saying, okay, give us a approval. So now suppose we give them approval, why they are coming here? Because it will be treated as a new class of drugs, and India is a huge market. So you have to understand that why not you develop whatever extracts are there. So now what was the thought process? See what ma'am has told you and what she has explained you, that what we should have the science-based drugs. A robust scientific data is to be there, which is to be supported. And you should have what? That we should have what? Separate and appropriate regulatory provisions. Because when we are having 60 to 80% of herbal botanicals are there, why not we should have a separate regulatory process? Okay, you cannot treat them. And suppose you want to have it a phytopharmaceutical, then of course you have to go according to that. Okay, so you can see this, and I am giving this which are marked, that the student must see these things and they should Google it, because these days it is very easy for you to Google. In our time we used to sit in the libraries from morning till evening. Now you can just Google and see that what all is there. So please go in for that if you want to really know about the phytopharmaceuticals. So herbal, it was, see all these things you know. There is nothing which I am telling you new, that it provides a superior benefit risk ratio, okay, and evidence based, but you should have a evidence behind it. That is very important. So what was the government initiative? In August 2008, they formed this committee, and I am sure that you know all these names very well. Please, if you don't know, you can search for them, that what they have done to the your pharmacy profession. So they all thought of it. And now what is the stimuli to develop phytopharmaceuticals? What is the stimuli? Why you want to develop a phytopharmaceuticals? Because it will provide what? Impetus to herbal based products. Because mostly they used to say, why I should work on it, the industry is not taking it, isn't it? And industry was also very apprehensive about taking it. They say there is no day-to-day, you cannot maintain the same quality and other things. So it will give impetus to herbal-based products, and these are an alternatives to NCEs. Now why it is an alternative to NCE? What is an NCE? What is an NCE? So why it is an alternative? So nowadays it is not that single target and single molecule. Now it is what? It is one target. You can have multiple, there are multiple receptors and multiple this thing. So it means that what? That you should have a combination. Even the doctors are prescribing 3 or 4, because they believe that okay, you cannot just have just one to treat you, you should have a combination. So this is already there in a plant. Because sometimes when we teach pharmacognosy, we say, okay, this plant is what? Anti-inflammatory, oxidant, hepatoprotective, anti-diabetic. So students wonder how come like one herb or one medicinal plant is having all the properties. Probably she's saying like this, and when they write the answers, you know what they do? Common things. All are hepatoprotective, anti-diabetic, antioxidant. It is not like that, because there are separate constituents which are responsible for that. Now I just give you a simple example of a wound healing process. Now when a wound is to be treated, what all constituent it should be there? It should be antimicrobial, it should be anti-inflammatory, plus it should have something which is for the wound healing purpose. So it is the same thing, okay? Now it means that what it will meet, it will unmet needs which are multi-component drugs may be ideal for multi-target diseases. So you can have many targets and that can be used. Then it also prohibits cross prescription, because Honorable Supreme Court judgment is that you cannot have 2 or 3 which are having cross prescription. So if you are giving one herb which is a phytopharmaceutical, it also avoids your cross judgment of prohibiting, that is the cross prescription. So now what is the definition? This you have to understand, that it is a purified, it is not a crude, it is a purified and standardized fraction with defined minimum 4 bioactive or phytochemical compounds, qualitatively and quantitatively assessed of an extract or of a medicinal plant or its part for internal or external use of human beings or animals. So it means that you cannot take it as a crude extract. You have to have a purified. Now what do you mean by purification? How you will purify it? Now suppose I take an extract and I want that it should be anti-diabetic, because it is mentioned in traditional drugs that it is anti-diabetic. Now I only want to target that for anti-diabetic. So what I will do? It is bioactivity guided fractionation you will do. So how you will do bioactivity guided fractionation? That is, you can have certain in vitro assays. There are very good in vitro assays which you can do, and you can bioactivity guided, right? So it means that that particular purified fraction is only having those constituents which are responsible for diabetes plus the other things which are associated with diabetes. Are you getting the point? Okay. So it means that 4 bioactive or phytochemical compounds, but they should be specific to a particular plant. They should be specific. 2 or 3 can be the common ones, as I have told you, that flavonoids will be there, tannins will be there, but there should be at least 1 or 2 which are specific to that particular medicinal plant. So now if you see, there are draft regulations of chapter 4 and 4a. That is why I am telling you to read all this. It is very important, because you will see that this phytopharmaceutical drug will not come under the same regulations as you have the ASU drugs. It is now different, okay, which will be accepted by all the people all around, but yes, with a robust scientific data. Okay, so now if you see what you are going to do, now generally you say, okay, I have this technology of making an extract. Now what are these technologies? Not conventional burn. You have what? Supercritical fluid extraction. You have green technologies. So use all the technologies which you have. Now you have all the, all the leverage to use the technologies which are there. Modern technologies, you can use those, and you can develop something like this, which is having what? High degree of characterization. This is very important, that it should have a high degree of characterization. Now from where this high degree of characterization will come? From the integrated approach, from all the modern analytical techniques, and you can use those techniques for developing an phytopharmaceutical or a purified fraction of the extract. So this is your draft regulation, which is current ASU and licensing system, and the draft regulations are not a mandatory provision that applies to ASU drugs in any way. Now ASU drugs are different, and phytopharmaceuticals are different. So you have to understand that what are the regulations in chapter 4 and 4a before you go in for the development of the phytopharmaceuticals. So this phytopharmaceutical proposal was given like this, and it was well understood that it will have a scientific evaluation and data generation, and it will give boost to research in the drug development. Now what are the important points which you have to take care? That the leads can have their origin not only in traditional medicines, not necessary, from anywhere, any herb across the world you can take it. It is not that you have to take the drugs which are there in, medicinal plants which are there in your pharmacopoeias. Okay, then you can see, because traditional medicine system, there are so many here being practiced, you can pick up any plant from any system and you can start with, and then to be mandatory evaluated for safety and toxicology, which is very important, because generally we see that all the herbal drugs, they say it is safe. No, it is not like that. It is not at all safe. So you need to give a safety data. The toxicology, so you are also involving toxicologist along with you, right? And then such mandate, and moreover, lines are similar to synthetic compound based drugs, that is, which synthetic chemists are making. It will work on the similar lines. It means that what? The phytopharmaceutical which you have developed is just like a modern medicine. So now if it is a modern medicine, who can prescribe? Who can prescribe this? A phytopharmaceutical drug can be prescribed by a modern physician. He can also prescribe. So already you are getting a very wide range. So if you develop something, you are having your own intellectual property right, and it will have a very wide market, because now the doctor, he can also prescribe the phytopharmaceutical which you have developed. Otherwise, a modern physician cannot prescribe which is given in ASU. Only an Ayurvedic physician and others only can prescribe. Though at the back end, the modern doctors are prescribing ASU drugs, but now if you develop a phytopharmaceutical, they can prescribe that also. So this is the status which I told you, that is, if you can see, there is a Drug Technical Advisory Board, DTAB. That also you please, student must look into that, what is that board and how it is going to help. So I'll just move on further. Yeah. Let me go into now what are the expected outcomes? Why you wanted to do this? Because national labs in India, scientist group and industrial research and development who have been working on botanical leads can look forward to take this route for getting market permissions, as ma'am has done it, isn't it? She has developed the curcumin, right? So you can have that, and then it promotes what? Innovations. Now what are the innovations? That new method of extraction, new method of analysis, all these are your innovations to study the pharmacokinetics and to see what is, to study the pharmacodynamics also. All this which you are studying in pharmaceutics, you need to do for a phytopharmaceutical drug. So it will encourage what? Extensive evaluation of medicinal plants, expand the use of herbal products, and it will also meet the therapeutics for unmet medical needs. Because till date, now the doctors have realized that they cannot cure anything. They cannot cure. They can only prevent it. They cannot cure, they can only prevent. And thinking of curing it, they were increasing the medical burden so much. They can only, they can only prevent it. And what was the first line of Ayurveda? Or what we think? That it is better to prevent than to cure. So, moving to the same lines, but with all the scientific validation. Because now they have all the techniques, so they would like to see, okay, show me the NMR, show me the HPTLC, show me that you have all the 4 markers in one extract or not, and where are the pharmacokinetics, where are the pharmacodynamics. They are going to just they want to have this data. Even we like to have this data when somebody submits an application of phytopharmaceutical at CDSCO. So we are doing the same thing. So what are the regulatory issues? That phytopharmaceuticals, they offer an easy approach to as overseas registration process is concerned. Now, once you say that it is a phytopharmaceutical and Indian authorities has approved it, you will have no regulatory problem all over the world. But if they come to us and they say, please take this as a phytopharmaceutical because it is already there in United States Pharmacopoeia, we say no, we cannot take it unless you tell us that what you have done to it to develop as a new phytopharmaceutical. So we have more stringent regulations for developing a phytopharmaceutical. So what is the constitution? As I have told you that in Ayush domain, what is there? Only the crude extracts. Okay? But in phytopharmaceuticals, it is a purified fraction. So this is just because, that students, they like a table where you can simply compare it. So I thought, let me show you the table that how you can differentiate the two. That is number of ingredients, single or more than one you can have in Ayurvedic medicine. But in phytopharmaceuticals, only one single plant. So, many people, they came say, no, can we add 2 plants? It is so difficult to even manage one plant because it is having so many constituents. You cannot, have mixture of 2, then you have to manage 8. And then from where you will do the pharmacokinetics and pharmacodynamics. So it is not that easy. So only one single plant, and then combination with metals or minerals is permitted. But here it is not permitted. Crude extract, purified fraction as a proprietary medicine, but it will be an allopathic drug. So if it is an allopathic drug, you have a huge market. You have a huge market, and then prescribed by allopathic physicians, no, yes, they can prescribe. Then regulatory requirement, I have told you that extensive, exhaustive scientific data is required, otherwise you cannot do it. And then preclinical requirement is not mandatory in Ayurvedic medicine, but it is here mandatory. Clinical requirement not mandatory, but here it is mandatory. And development is 12 to 18 months in case of, but here you have to 4 or 5 years. Otherwise, how much time it takes to bring a new molecule? Ma'am just told you how much time it takes. So many years it takes. But already you are having something, why not to develop in 4 to 5 years? Because you are already having that plethora, you know, of all the medicinal plants are there, the crude extracts are there, which have been worked very widely across. Pick up any crude extract. Now you know that standardization has also been done. Select your target and you can develop a phytopharmaceutical. Yes, but with the help of your 4 departments together. So already it is what? It is a holistic and it is an integrated approach. So holistic approach, I think in between I have already explained you, because you have been using all these techniques. So I will, one more thing I would like to tell you, that the correlation of botanical plant integrity, good collection practices, all these good collection practices, botanical reference material, chemistry manufacturing control of the enriched fraction, GMP with at least 4 marker compounds, all these things which you do it in your modern medicine, or when you take one constituent which you do, you have to take care of all these things which are already there in pharmaceutics. So if a person is there in pharmacognosy and phytochemistry, he is actually working much, much more, you know, because you are taking out something from the plant. Here you have been given just one purified, isolated, which has all the assay procedures given in the pharmacopoeias. So it is not that difficult. Yes, it is difficult to work on medicinal plants. It is not easy job. So in vitro activity, in vivo activity, as I told you, GMP preparation, safety pharmacology, which you are doing, and then INT package, that all has to be given with this. So what are the key measures? That you have to follow comparable regulatory guidelines as conventional drugs, and evidence-based phytopharmaceutical should be there, and it should contain pharmacologically active ingredients that interact with the protein structures. Now why drug targets? You know, because all your drug targets, receptors, you must be studying in pharmacology, they are all proteins, isn't it? That is why, you know, proteomics has upper hand than genomics, because it is very difficult to find out the whole genome than to have the proteomics, because they are counted number of proteins, and anything which happens inside the body can be checked by proteomics. If the disease is there, what is upregulation, downregulation, or if you have given a medicine, what is happening to that. So this you have to understand. So what is the way forward? That is pharmacological activity and dose dependence, active ingredients, then determine the target before, and go in for bio-guided extraction. So what you have to do, you have to learn the assay procedures which are there to check the anti-diabetic activity. Can you name any 2 assays which are for anti-diabetic activity? Any 2 assays which are very simple. You suppose you are having a crude extract, you can go on doing the fractionation and do those in vitro assays. So it means that you have to also learn all the in vitro assays which are now available, and then that I have, I think, already described. So what are the initiatives which you have to take? That the need of the hour is what? To bring reputed herbal drugs. To bring reputed herbal drugs under the umbrella of phytopharmaceuticals. Why? Because your future is indigenous. You don't have to run here and there thinking of I should take the molecule from there or take the molecule from there. Future is indigenous. You are already having all the extracts and you need to work on it. Okay? So thank you for collective learning. Thank you so much.
Thank you, ma'am, for guiding and updating our knowledge on the traditional approaches of medicine and innovations related to it. Now, I would like to request Dr. R.A. Patil ma'am to kindly summarize the session. Please ma'am.
Thank you so much, ma'am. Thank you so much for briefing us phytopharmaceutical, such a vast topic in just 1 hour. Those who are working with phytopharmaceuticals or medicinal plants, they do know that how difficult it is to work with the medicinal plants, herbal plants, or this phytoconstituents. Okay, because as we say that herbal plants, medicines, they are like a group of compounds, and it becomes really difficult to isolate them, to study them. So this concept of phytopharmaceuticals, definitely right now it is of point of concern, because many of us, we are using this medicinal plants or herbal medicines, Ayurvedic products in a large scale nowadays, instead of allopathic medicines, we must say that. And their concern that is also increased, but many times we are unaware about the side effects of those plants, those medicines, because as everything that has been taken for long time or in large quantity has some adverse effects. So the concern should be important, I think. And ma'am very nicely explained all these things in just 1 hour, and she summarized so many things that you can utilize it for your future's research work as well. Thank you so much, ma'am. And it's my pleasure to chair your session, ma'am. Thank you.
Thank you, ma'am. From the participants, if anyone has any question, you may ask. Any questions from the participants? Thank you, ma'am. I would like to request Dr. R.A. Patil ma'am to present Dr. Vidhu Eri ma'am a certificate and memento as a token of gratitude. Please, ma'am. Thank you, ma'am. Now I would like to request Dr. Vidhu Eri ma'am to present Dr. R.A. Patil ma'am with a certificate and memento as a token of gratitude. Please, ma'am. Thank you, ma'am. Here I conclude that the second session is over. Thank you, everyone. I request all to please proceed for lunch and please be present here by 3 p.m. sharp. So please be present here